สวัสดีครับ Good afternoon, everyone. Welcome back to the afternoon sessions. We do hope that everyone enjoy your lunch. In this morning sessions, we have 6 speakers. For those who just come to join our meeting, this is the 3rd Veterinary Technology and Nursing International Seminar, Engaging of Science for Animal Health Innovations. This afternoon session's theme is Innovative Animal Health. We have 13 speakers. Firstly, I'm pleased to invite Professor Dr. Serge Moran and Assistant Professor Dr. Laksa to be moderator to these sessions. Please. So the participants who just come to join our meetings, you can also registration form was registered from our registration form in our website www.vettech.ku.ac.th. The scheduled programs also posted in that website. Okay, now the moderator is also ready. Please.
Thank you. Welcome everyone back to our afternoon session. In the afternoon, we will have another 13 speakers. First speaker, I will start with the first speaker. First speaker is Dr. Michel Karin Vichativsky. He's from Greece, KU, Sirat, France, and he will present in the topic Socio-Ecology of Dog-borne zoonotic diseases in Southeast Asia. Please welcome.
And I will have the pleasure to facilitate the session on the dog-borne zoonotic diseases this afternoon, and to give you a first presentation about a project that we have been carrying out for the past 2 years on specifically this topic about dog-borne zoonotic diseases. So projects, a few words about the context, why we're interested in dogs in Asia. Well, first of all, as you know, domestic dogs have had a very long-term relationship and very intimate relationship with humans. They were the first domesticated animal, and one of the centers of the domestication was in Asia. And currently, there are a very abundant and widespread population of free-roaming dogs, especially in Southeast Asia. And during this very long relationship, dogs have acquired a very special place in human societies with social, cultural, religious, and also more practical and management questions, and they have established very intimate relationships with people, and especially at the very core of millions of households around the world and in Asia. Dogs are also a concern because they actually harbor a number of zoonotic diseases, which are currently a major public health concern, especially in Southeast Asia. Rabies and a number of neglected zoonotic diseases that are currently a concern. Having said that, dogs are very interesting from an epidemiological point of view because they may serve as a bridge, a link between wildlife and natural habitats, or even man-transformed habitats, where they roam often freely or accompanying their human host, and humans, so they may serve as a bridge host. And dogs may also be used as a sentinel to monitor the occurrence of pathogens from vectors or the environment that may be emerging pathogens. So because of all these points, there is clearly a need to address epidemiology of these diseases through a social ecological approach, interdisciplinary approach. And this is what we have adopted as part of the CDOC project, which stands for Social Ecological Approach of Dog-Borne Zoonotic Disease in Southeast Asia. The project associates CIRAD for France, Indonesia with University Gajah Mada, and Belgium with the University of Liège. Also associated are Cambodia with the Institut Pasteur of Cambodia, and Thailand with Casings University and Maidong University. So the activities, research activities have been implemented and will be implemented in 3 sites in Indonesia, Cambodia, and Thailand. So the project is structured around several work packages. One is focusing on understanding the social and anthropological aspects of dog ownership, the perceptions by owners and other stakeholders about the dogs, the risk they may represent. The second work package is focusing on dog ecology, and especially analyzing the movements of the dogs at the scale of a landscape, including the village and neighboring habitats, and also looking at population dynamics of the dogs. And a third package is addressing measuring, estimating the seroprevalence and of several dog-borne zoonotic diseases, and I will give more details in the next slides about which pathogens we are interested in. So the ambition is also to link these 3 different aspects to better understand how dog distribution, the contact networks they establish can allow us to better understand the epidemiology of these dog-borne diseases and their potential roles as sentinels or bridge hosts, as I've just explained before. So I will not have the time today to give you some illustration about all the work packages and the different methods that we've been using. For the dog ecology and especially the movement surveys, you will have a presentation after this one by Prof. Anand Pompoen from Casings University, who will be giving you more details about the performance of the GPS collars that were developed and the kind of data that we are expecting to do, and especially the preliminary work that has been done with colleagues and a master's student, Céline Sin from Montpellier. So that will be the next presentation. And then you will have an illustration of some results about leptospirosis for zoonotic disease prevalence by Dr. Putukri from University Gajah Mada. On my side, I will just take this opportunity to give you some preliminary results about one specific activity on dog ecology and especially population dynamics using a protocol we developed on camera traps. So the objective of this activity is to first, through a participatory approach, decide with the local communities, the villagers and other stakeholders, where we can set these camera traps that would be recording automatically all the objects or animals that would be passing in front of the camera traps. So we first have this participatory workshop where we decide first the geographic distribution of the camera traps, but also a specific hypothesis where the villagers believe that there are higher densities of dogs or resources that are associated with the dogs where we may have a greater chance of capturing dog pictures, such as the one you can see at the bottom of this slide. In order to do that and to analyze the data, we've used artificial intelligence and deep learning neural network, and we trained the neural network to identify the images of the dogs. And this is a short illustration showing on the right the algorithm was trained with a dog-specific database. So you can see that the confidence of identifying a dog is always and constantly very high. But on the left, it was the same pictures, but analyzed and trained with a database that is much more generalist. And you can see that the dog is mistaken for other things like dog, horse, cow, or whatever. So that shows the importance of this initial phase of training the algorithm with relevant data sets. On the next slide is to illustrate the fact that one of the issues is that we want to identify all the dogs in the pictures. So on the left, these two dogs that are present are correctly identified. And on the right pictures, you can see that the dog is actually included in and merged with the object, which is in this case a motorbike, and disappears. So this one would be called as a false negative because it will say that there is no dog where there is actually a dog. On this picture, you can also see that we successfully implemented a procedure to blur the faces of people to make sure that all the data the pictures we analyze are anonymous, so that we don't get any personal information out of these. And this is the same also for the number plate of the motorbike, which is also blurred. So that was the first aim that we had, which was first to select pictures with dogs and remove all potentially identifiable features like faces and text. So we applied that on one specific recording session, which consisted in 3 nights and 3 days of recording, continuous recording, using 14 camera traps in a village in Indonesia. And you can see that the amount of pictures is huge, 32,000 pictures. And when we applied this algorithm, the automated procedure, a lot of pictures were recording without dog, nearly half of them, whereas the rest, most of the pictures, at least one dog was supposed to be identified. Most of the time, a unique dog was identified, but as seen in this picture, some of them may have been missed, but other ones may have been actually recorded where there was no dog, and I will show you these cases of false positive, whereas the false negative one was the previous picture. So altogether, a lot of the majority of the pictures selected had no dogs, and most of them we had to remove the faces and all the other identifiable items from the pictures. So we validated the performance of this procedure, this automatic procedure with the artificial intelligence using a data set of manually examined picture where we manually spotted each dog and person. And altogether, the algorithm was performing pretty well. We were most of them, more than 90% of the time, in agreement where on the pictures where there were no dogs, with a few false negative, but 5%, that's fairly good. But with this particular training using the dog database, we ended up with a lot of false positive. You can see on this picture on the left that the algorithm was identifying one dog on top of this roof, which obviously was not the case. So this is the next step is to improve this detection of false positive individual false positive dogs on the pictures. So the next step will be also so to improve the performance of the algorithm, and also the ambition is really to make sure that we can recognize individual dogs. We want to use this data set to assess the distribution of the dogs. The same dog, if we can recognize it, may be recognized on different camera traps, which will allow us to have an estimation of the range and the frequency of co-occurrence of the dogs. But also, this type of data sets using several sessions of recording may be used for capture mark recapture analysis, which will give us some insight, not only an estimation of the population of the dogs, but also the population dynamics, which is what we want to do. And there will be a master student that will be doing his master work on this topic next year. So that's it for the very short illustration of some of the work that we've been doing as part of CDOXI work. You will have more presentations by, as I mentioned, by Prof. Annan and Dr. Putu. And you will have also, after your third presentation, on work that we intend to carry out next year on the screening of arboviruses in both dogs and dog owners. And there will be a presentation on the methods by Dr. Heidi from Institut Pasteur of Cambodia. And then the last session, the last presentation of this session will be by Dr. Johan Michaud from the University of Liège, who will give us insights about how we can use microbiome analysis as part of CDOXI project. So without further delaying, I will thank you for your attention, and I'd like to thank the organizer of this seminar and also all the numerous collaborators who participated in this work. Thank you. Okay, good afternoon. This was the video, and now I can interact live, although we are not all at the same place. I think we've got plenty of time for questions, clarification. I realized the previous video that was recorded may not have been completely clear. So if you've got any clarification questions about the work that we've been doing, small work about the dog-associated diseases, I would be very happy to try to answer them. So I don't see anything on the chat for that. Would you have any hands raised? Okay, maybe I can go back to one of the presentations that were made to clarify exactly first of all, the rationale, why we are interested in dog-borne diseases. We are all in the group very interested in the wild-domestic interface. And when considering this interaction, emerging diseases between wildlife, livestock, human, that's when we realized really that dogs play a very special role, and that's why we concentrated our efforts on that. So maybe to explain a bit more about this role about bridge species, this is something that theoretically has been emphasized, so showing that some species may not be very important in maintaining the diseases, but may play a crucial role in transferring from one host to the other one. And if we look from this perspective at dogs, they are really very interesting because they interact directly with the wild habitats and the wildlife. So potentially dogs may scavenge or maybe hunting some rodents or anything in the habitats, and then may be infected. And although they may not be able alone to maintain some of the pathogens, as they also interact with very intimately with their dog owners, they may play this role of transferring the pathogens directly to the human host. And that's why we started concentrating really our interest on these dog-borne diseases and realizing that it required not only classical epidemiological follow-up, but also clearly including also anthropological and social dimension, because dogs are animals, of course, but they are owned and they've got very special relationship, especially with kids, with children, but also they are used for several activities. And that's why there was really this need for social and anthropological lens, understanding better how the community perceive the dogs, how they manage them. And so that was the first component. And then the second component is really look at dogs like using ecology techniques and looking at the ecology of dogs in the free-roaming dogs as with similar techniques as what we would be using also for wildlife. And that's where this idea of dogs emerged and the CDOC project emerged. I don't see questions. Are there some in the chat?
So if there's no question, I would like to say thank you to Dr. Michel. And we will move to the next speaker. Our next presenter is Professor Anand Ponperm. He's from Faculty of Computer Engineering, เกษตรศาสตร์ University, Thailand. He will present in the topic Tracking Movements of Dogs in Rural Landscape of Southeast Asia. Please welcome Professor Anand.
Okay. Good afternoon, everyone. Can you hear me? Okay, all right. Okay, let me share my slide. Sorry. Sharing, presenting. I should change into, let me move to share. Just one moment. Okay, can you see my slide now? Hello? Yes?
Can.
Okay, all right. Okay, let me start. Okay, let me start this one. So the project of tracking movements of dogs in rural landscape of Southeast Asia, normally I and my team is just this one part of the big team of the Dr. Michel, Dr. Serge. Okay, so we are just a technical team for doing the tracking device. Okay, so for today, I'm gonna talk a little bit about the tracking device, what we are using, what we implemented, and also what is the ongoing research that we are doing for tracking the dogs. Okay, so first of all, okay, so our tracking systems. So tracking system will have two. Actually, there's two subsystems. The first one called data locker, and another one called real-time tracking here. The first one, the first system we call a data locker because they are only locking the location of the dog without sending anything out from the tracking device. We just track the location and keep it at the dog collar here. The second one is the one that to make it real time or near real time tracking by sending the information or the tracking to the server. Obviously, they're gonna track and keep all the record in the collar as well. But they do the second part is to sending it online. Okay. So this is two part of two system that we're gonna use in the tracking the dog that we implemented. Okay. The system is gonna be like this one. For the system overview, you can see that they have two types. The first, the number one and number two. Number one is just a standalone collar. That is, we install the tracking device with the dog, okay, and let them go running around. And then after the periods of interesting, for example, a month or two or something, we're gonna collect the device back by taking the dog, okay, and take the collar. For the second type is the number two. You see the number two here is the collar that can send the information directly to the gateway. So the gateway is composed of two parts. The first part is for communication to the dog collars. This part we use a LoRa system. It is the long range, low power. Okay, so we can keep the system working for a little bit long than the regular Wi-Fi that you guys using. Okay, so this part is going to be upload all the information to the controllers. This one is communicated by LoRa, and then we're going to have a pocket Wi-Fi for connect all this data and transmit this one to the server. This over the internet. It means that we are install the collars in non-province, and after that we're gonna use we're gonna view it in Bangkok. It is fine. Okay, so this is the type of the two collar system that we deploy. This one is a node component. There are two types. The first one, right? The first one is only tracking. So we can have the module for keeping the GPS and the module for locking. Okay, so only only two layers here. If you see from this slide, we have three layers. The first two layers is gonna be for the tracking device that keep only data locker only. For the second type, we have three, right? We're gonna have the last one, or LoRa, the radio communication here. We use the 433 megahertz for sending the information from the nodes to the server. Okay. This one, the component, if we go dig a little bit in component, please bear with me because you guys are wet, right? So we are engineers. So I just try to not to go too deep in the technical details about this kind of technical issue here. So the node is going to be some microprocessors that we use and put in the LoRa module. And we have the
teacher.
Yeah. Okay.
In the first exam, can I copy
just one
ตาราง
moment.
นะครับ ผมสามารถก๊อปตารางได้ไหมครับ
Chandom just answer that. Yes.
Okay, thank you.
Okay, just sorry. Okay, sorry for interruption. Okay, so the node is gonna be microprocessors and LoRa communication here. Okay, so for the gateway, they're gonna have a microprocessors and the LoRa module. Okay, I just make a parenthesis here, 4 channels. So it means that we're gonna have the upgrade one. I'm gonna show you soon, and Raspberry Pi and pocket Wi-Fi here. Okay. For the node casings, the thing that we used to have is the node. So this is the size of the node. If you can see from number one here, this data lockers offline is going to be a little bit smaller because it's only 2 layers or 2 stacks. And for the number 2, the real-time tracking online is going to be 3 layers. It's going to be stacked a little bit bigger, but the size itself is going to be, as you see by the rulers, about 7 centimeters. So that size is not that too big. Okay, the batteries that we use is going to be 3000 milliamp hour here, so it's going to be a little bit small. The reason that we make this small because we want to keep the weight of our collar to be less than 5% of the dog weight. Okay, so we try to keep it very minimal to not interfere with the dog life here. So this is the new node casing here. So we try to put a casing here and then try to seal, because last time that we deployed in Nan province, we have found about 10% of node that has been damaged because the dog is going to the water. Okay, or maybe it's rain or something, it's a short circuit. So there has some part that is a little bit not tolerate to the water here. So we try to make it more robust in the sense of the new casing here. The gateway, this is the main part that we changed from the last deployment from the gateway version 1. If you can see from this one, we're gonna have the Lola module, the last belly part, and make it this one connect to the collar. However, for the new one, let me show you the new one here. This is the new one here. We have the second version. So we now upgrade the communication of the LoRa to become a 4 channels. Okay, instead of 1, normally it's only 1, and now we use these 4 channels. So this 4 channels mean that we can install a lot of collars at the and let them sense the data at the same time without collision. So it means that if we deploy a little bit big groups of dogs, for example, 20, 30 at one place, the gateway can collect all the data as simultaneously here. We're going to use the antenna. You see these 4 antennas because it's a 4 transceiver. So we can set 4 channel for communicate to the dog collars here. Okay. This is the pictures that you've seen before for the helping of the vet for catch the dog and try to install, and we try to install the collar to the dog. So the left hand side is the collar look like, and we install the dog in the right hand side here. Okay. For the gateway, at the gateway is required electricity. So we need to install for some of the place that have electricity available here. We also sometimes we can do, if they are very remote, we can install the maybe solar systems. Okay, but in our case we not see that one yet. Okay, we just only use the electricities of the villagers. Okay. This is the real-time tracking version 1 that we used last year. So you can see that the name of the dog and the colors is the represent that the dog is appears or not. For example, the green one here, the green one number 13 here, the dog's name Kiao. So his Kiao is appear for a while here, but number 9, the red one here is disappear for a while, something like that. So we can see that the dog is coming back and forth, but it still cannot see a lot of tracking or tracing in real time here. Okay. Therefore, what is the next system that we are working on? So there are 2 things that we are working on now. Is the first one we call behavior tracking here, and the second one is the tracking platform. Let's go for the first one, for the behavior tracking here. If you see the system here, behavior tracking, what does it mean? It means that if we have a dog and we see that the dog is not moving at all, are they sick? Are they eating? Are they stand up? Or they are moving around? Are they praying? Something like that. So if we do know some of the situation or the status of the dog, maybe can help analysis for the result, better result for this case. So in the tracking device, the new module, we are not using that yet. We are installed this one now in the lab and the testing. We're gonna use the accelerometers here. This one, the accelerometer is going to be sensing all the movement of the dog and then send the LoRa, send through the LoRa to the gateway here. And the gateway is going to be collect all the information and send to the server. At this gateway, it can be many things. We can just relay all the information, this one, and send to the server. Or another way around, we can just calculate and do some of the adjustments, or we call it a Fox computing. So we do it the gateway, compute something, summarize something, is the dog is sitting or standing, something like that. So we can summarize the data before sending to the internet as well. The implementation we testing in the lab is going to be look like this one. We can have some sample dogs. Okay, well, it's one of my students. Okay, Kamon Chinok here. So they are just study here. So they will use the accelerometer board, MPU, this one, and then they install in the collar, a small collar one, and then put it in the dog like that one. This one we install with very limited batteries. So I make it small because the dog, this one is very small size here. Okay, so from this one, if we tracking this one, we can see that once the data has been sent, it's going to be show your location, the GPS, and also some of the information about the movement of the dog. Okay, this is maybe they're going to stand up, they're going to be going down, slowing down like that one. However, after the testing results show, it is going to be not really accurate. It's going to be about 60-70% that can guess the gesture of the dog correctly. Okay. However, we are still a testing phase, and maybe we need to have more device, not only the accelerometers. We might have a gyrometers or something that use for tracking and see the movement of the dog or the head of the dog. Okay. So that is the first part that we are doing ongoing research here. The second part is after we are tracking. If you see from the last slide that we have a dot, and show that this is the green dot, this dog is here, this is the red dot, the dog is has been disappeared for a while or something like that. The new tracking platform that we are working on is gonna be like that. It's like a map. So we can send the data information, send back to the server, and our server gonna be clear the platform and doing some API, application program interface, and then showing the location on the map. For example, on the left-hand side, I show that for by 1 minute, every minute we testing. For every minute they're sending the data, and then we can see the location of the dog at the movement. Or in 10 minutes, we can see more roughly, something like that. Because sometimes it depends on the scaling that you are, or resolution that you want to monitor. If you want a movement in 1 day and you want to see every minute, so like that one, so we can trace. However, it's going to be a trade-off between how many times that you sense the data, sensing the location and send the data back, and how long you want the system to be last. Okay, so this is going to be a trade-off here. So this is the second part that we are working on. We want something to become a tracking platform. Next time, if we install the new collar to the dog, we should be implement very minimally, and then that device of the collar should be sent the data directly to the server, and the one who manage the system can see or view the tracking or the tracing of the dog directly here. Okay, so that could be it for my presentation. Any questions? So I try to make it 10-15 minutes, right? I see Michelle is smiling.
Thank you, Profan. I think the moderator may be seeing some questions. If they are not.
It's time for question. Please feel free to ask the question by yourself, or you can send the message in the chat box. There's one question. Can this tracking system use with the cat?
Normally okay, let me answer for that one. So normally the tracking systems can be added to any kinds of animals that a little bit not smaller than the cat or the dog, because the size is, the weight is around 300 grams. So the animals that you want to track, it should be about less than 10 kilos, about 5 to 10 kilos is to be fine. So the cat is in the sense that it should be still okay, but it depends on the battery that you use. If you use a large battery size, it's gonna be over the 5% weight of the cat. If the animal is smaller than that, I don't recommend for using this system here. Okay. We do implement this one for a cow, for the cattle. That one is a little bit easier because the cow is quite big and the size is big, so we can add more battery size to that one.
Unless there are other questions, maybe I would like to give a clarification about what important point that we were giving, which is the frequency at which we want to record the points and the location of the dogs. In fact, in our case, for the epidemiology and ecology of the dogs, there would be 3 different questions that we would be trying to answer. The first one would be to look at the home range of the dogs, and especially understand what are their habitat use, how long they spend in natural forest or in the urban area or in the crop fields or all these. And this, it only makes sense to try to evaluate that over a long period, 1 month, 6 months, or even 1 year, if we look at seasonal variations. So for this, you do not need to have the frequency of the points taken as frequently. But there are other questions like, are the dogs moving from one side to the other one independently, so walking or maybe running in some occasions, or are they carried, in the motorbike or in the car or with their owners? And that's to understand and to monitor that, we would need to have much more frequent data points. And that's why the compromise between the number, the frequency of how often we take the points and how much battery we use, for recording these, it's always a kind of trade-off that we are making. So maybe to explain to the colleagues. And the third question we are interested in is the contact patterns. And in fact, measuring the contact when we've got 2 dogs that have got GPS collars, we also need to have quite frequent GPS locations so that we are able to say, yes, the dogs were at the same place at the same time. And if you've got a lag that is more than 1 hour and so on, then you cannot assess that very accurately. So that's why this question of the frequency of the data point collection is so important, but it's really depending on the specific question, the research question for epidemiology or ecology that we are trying to answer. Okay, more questions, comments?
Questions?
We still have a lot of work for Fanon. That's good news.
So we are gonna go to Nans province, right? On this Sunday.
Exactly. We're starting Sunday for the next session.
Okay. All right.
No more question. Thank you
right.
very much, Professor Anand.
Okay, thanks for all you guys. Okay.
Okay, we will move to our next presenter. Our next presenter is Dr. Heri Wijayanto. He is from One Health Collaborating Center, University of Gajah, Indonesia. His topic is the relationship between the dog's home range and zoonotic disease transmission in Karangasem, Bali. Please welcome Dr. Henry.
Thank you, moderator. Hope you all in very good conditions, and it's my pleasure to present our result of the field surveillance as part of the CDOXY project. And we did our surveillance in Karangasem, Bali, in Sumatran district, and the village is Banjar village in Banjar village, there are a lot of dogs that freely ranging from the corner of the village to the other corner of the village, and the village itself from the most outer border to the other outer borders is about 2 kilometers. Before we enter to the surveillance result, we would like to introducing some emerging infectious diseases happen around the world from 1940 until 2000, and all of the disease was associated with animals. And if you look at the second figure, we can see that the white block is representing wildlife animals who are contributing to the emerging infectious disease. And we did our surveillance in Tibetan because Tibetan before is the red zone village for rabies outbreak, but now the rabies is already mitigated by the local government. And surprisingly, during our surveillance, we also found some leptospirosis happen in the dogs. Okay, this is the place where we did our surveillance in the most beautiful islands, Bali. And this is the Sibutan village. The green line is the border of the village, and the outer part of the border have about 2 kilometer distance. To record the activities of the dogs, we put collar in some dogs, and from 35 dogs, we did the blood sampling. 12 dogs out of the 35 dogs ranging with GPS collar on the necks. And during a week, we record the area of the dogs, not only territory, also home range. And from that surveillance, we found 7 dogs out of the 35 dogs suffering leptospirosis. And from the 7 dogs who suffering leptospirosis, 4 of the dogs are the dogs with collar on the necks. And this is home of the dogs who positive with leptospira. And all of these dogs have home range that representing by the red, green, and blue line. I will not focusing on the leptospira itself, but I would like to highlight how the disease is transmitted from dogs to dogs and from dogs to the owners. Okay, let's see. Each dog have home range, and each home range have interception area. Look at this area, this one, and this area. And all of the dogs have meeting point. This is the first meeting point, and then this is the second meeting point, and this is the third meeting point of the dogs. And all of the meeting point actually located in the interceptions area among the dogs. And if one of the dogs suffering leptospira, we can imagine that this dog can be transmitting leptospirosis into other dogs, and the other dogs also will be can transfer or transmit the leptospira to other dogs. Because if we look at this point, actually why they gathering in this site, because this is the site where the human activities is very high. This site is where the locations close to the traditional shop, and many people usually sitting together and talk in the shop and throw away part of the food and the dog usually just eating the food from the human. This also, and this is also temple where usually many people come and brings many food for ritual processions. And after that, they will throw away the food and the dogs will be accept the food. So that's why we can understand now that the human activities also will be related with dogs, and the dogs itself will be food dependence into human being. And because this close relationships, the spillover of the disease from dogs to human being is very, very potential. This is all the examples of how the disease spill over from animals into human being, because we know that each dog will be put their urines to identify their territory and their home range. And in Sibetan, there are many salak fruit tree in Sibetan, and we know that salak fruit or snake fruit is located in the bottom of the tree, close to the ground. And if the dogs put their urine, it will be contaminating the salak fruit, and the salak fruit will be touched by human being. Not only that, surrounding the house, the dogs also will put urines, maybe in the corner, maybe in the yard, or maybe in the pets, and so on. And this is Leptospira study in Brazil. And this is also showing to us how the dogs very, very potential or have contributions to transmitting the disease into human being. So as the conclusion of my presentation, we can conclude that not only dog actually, but in this case, we use dogs as the target of surveillance, and the dogs in Sibetan have an important role in Leptospira species transmissions to human through the animal behavior, and human-animal interactions contributes to the zoonotic disease transmissions. Okay, thank you very much for your kind attentions. And this is I show you our teams in Sidoksi, Bali. And if we look at this picture, no one wearing a mask because we did the surveillance in this figure before coronavirus pandemic, and this is during coronavirus pandemic. And this is all of our team. That's my presentation. So let's have a very productive discussion afterward. Thank you very much. My name is Harry from Gajah Mada University. Thank you. Any question, please?
Are there any questions from the floor, or you can text message into the chat box?
It looks like no questions. Maybe myself.
Yeah, I think your speech was very clear. One question, is there any reason to think that leptospirosis may be a bigger problem in Sibetan compared to Bali, and Bali compared to the other islands, such as Java, for instance? If you look at the previous records of epidemics?
Yeah, thank you for the question, Michelle. Actually, leptospirosis is one of the neglected diseases spread out from the animals into human. But in fact, every year we have a victim of human because Leptospira. Sometimes because they are maybe they are not realized that that is leptospirosis, and when they realize that is severe diseases and come to the hospital, usually already late. So even not only in a remote area, even in the urban area like Yogyakarta, every year we have a victim of leptospirosis because too late to treat in the hospital. Because you know that leptospirosis usually will be attached to the nephron in the ren and also liver. And even my neighbor, about 2 years ago, 2 of my neighbors suffering leptospirosis, and one of them passed away because too late. And the first one, fortunately, we can save the other one, but must be receive hemodialysis about 10 times before recover from the leptospirosis. Yeah, this is maybe a little bit ironic because Leptospira still have victim in every years, but this include in one neglected disease from the animals. Okay, Michelle.
Thank you. Yeah, that's one example where it's not just the role, epidemiological role that the dogs may be playing, because leptospirosis will have different epidemiological cycles according to the conditions and the hosts that are present and so on. But that may be another example where we may use dogs as sentinels of the risk that the owners may be having in contracting the disease. And that's a good example, and there will be other presentation just after where we try to also investigate that. It's not just the role, epidemiological role that the dogs may be having, but it's also, can we use them as sentinels of the risk that the owners are having of contracting the several diseases or zoonotic diseases, which is slightly different than this notion of reservoir, where the hosts should be able to maintain and transmit the disease. In that case, whether it has effects on the dog themselves is not really the question we are looking at, but whether they can be reliable indicators or proxies of the risk that may be threatening the dogs, their owners.
Yep. So I think since we found some cases of Leptospira in the dogs, I think this is one of the alarm from the for the people surrounding the village, because not only dogs, like in Jogja, leptospirosis mostly spreading by the rat, because not too much dogs in Jogja, and if we found the dogs, usually the dogs have a good owners. There is very little street dog in Yogyakarta. So usually leptospirosis come from the rat in Jogja. Okay, yeah, that's right, Michelle. So the dogs, we can put the dogs as the sentinels, and if the dogs positive of leptospira, so this become an alarm into the people surround the village.
That's really the philosophy, the One Health dog, healthy communities. That's
yep. Right. Thank you.
So use message to the chat box. If have no questions, may I move to the next speaker?
Okay. Thank you very much.
Thank you very much, Dr. Haley.
Yeah.
The next speaker will be Dr. Haiti. Harvard. He's from Institute Pasteur of Cambodia, Virology, Cambodia. He gonna present in the topics of screening for arbovirus in dogs and humans using NT assay insight from Cambodia. Please.
Hello, everybody. I'm Heidi Auerswald from the Institute Pasteur in Cambodia. I'd like to talk about the arbovirus neutralization platform that we have here that we used for several seroprevalence studies in dogs and in humans. I'd like to emphasize that virus neutralization antibodies are quite interesting to measure because they are not only binding to the virus, what you would measure, for example, with an ELISA, but they're also functional. So they're really involved in the blocking of the viral infection. This can happen through several stages. So the antibodies bind to the virus and therefore can block the virus attachment itself. They also can block the endocytosis of viral particles into the cell, and they also could block the encoding of the virion so that the viral RNA cannot be released into the cell, and therefore the blocking of the replication start of the virus is blocked. Furthermore, I'd like to go into more technical details because I know some people are thinking about setting up a neutralization assay for one or several viruses. Look in for real. So it's quite an extensive method where you have to have a well-run cell culture as well as virus culture. And of course, because we are working with live viruses, you have to have the laboratory concerning your biosafety level. So for a lot of arboviruses in an endemic area, this would be a BSL-2 laboratory. However, if you work with more pathogenic and novel viruses, you might need a BSL-3 laboratory. So what else do you need to set up an NT? You need, of course, your positive and negative controls. This is like for all serological assays, a very important thing to have so that you always can confirm that your results that you obtain are true. You need the arbovirus of interest. You can either use a prototype strain, which is most of the time the first strain that was isolated, or the most the first ancestor of a certain virus. However, often it's also very interesting to check, especially in a local population, the immune response against local isolates. Of course, you also need cells, and don't underestimate the amount of time and experience you need for neutralization, especially in the setup phase. What also comes in quite handy is if you have certain equipment to simplify or automate the readout. I will go into that a little bit further later. So how does the workflow look like for neutralization assay? You have your patient serum or plasma, and this you dilute in a serial dilution series. You do the same with your positive and negative control. Then you add a constant amount of virus. So to every serial dilution of serum, you add the same amount of virus. And this is then the neutralization step. So if there are antibodies in your serum, then they will neutralizing antibodies that are specific to the virus that you added. Then these antibodies will bind to the virus and cause a so-called neutralization reaction. This is normally something that you do for 1 hour at 37 degrees. You also can do it longer to really make sure that the neutralization is happening so that the antibodies really bind. However, be aware, because you do it at 37 degrees, not a lot of viruses are stable for long under these conditions because there are no cells involved in this step. So it's kind of a balancing act that sometimes have to be optimized for each virus if you do only a neutralization for 30 minutes or for 1 hour or even longer. After this neutralization step, you add this mixture to cells. We are using adherent cells, and we use a nearly 100% covalent monolayer, but also the cell density is something that is an obstacle for optimization. So you have to adapt the density and also the cells you're using to the virus you're using. This is also a step that can range between 30 minutes to up to 2 hours. And this is the absorption step. So virus that is unbound, so that is still free, will get attached to the cells, and later it can then infect the cell. So after this absorption step, you will replace the serum virus mix. So you simply take it off and you replace it by a semi-solid overlay medium. This is very important because most of the neutralization assays, they measure infection events and not into a single cell. They measure infection events for multiple rounds of infection. That means you first infect one cell, but then you wait several days so that the infection can kind of infect the neighboring cells. But if you would have a liquid overlay medium, then the new virus from this initially infected cell would simply be released into the liquid medium and then can infect any cell in the plate. But this is what you want to prevent. Therefore, you create kind of a barrier. So you don't use a liquid medium, you use a semi-solid medium. A lot of people use agarose. I prefer CMC because then I don't need a microwave. But this is like a technical detail that is can be optimized also for each virus. And also sometimes some cells like agarose more than CMC. So this is a tricky part that you can optimize quite a while around. So then you let your virus infect the cell for several times, for several days most of the times. There are some really fast viruses, especially alpha viruses, where it might be that it's only like 12 hours or 16 hours. So again, this is a step you have to optimize regarding your virus in your cells. And at the end of your infection period, you have then a readout that can be very different depending on your equipment and also what kind of neutralization assay you performed. This is how a plaque or foci formation looks like. So it's, in principle, you have an initial infection and then you see, due to the semi-solid overlay, that the viral infection spreads to the neighboring cells and not randomly across the plate. This is exactly what we want to see because then we can quantify the initial infection events, which is not always only one cell, but a foci of infection or a plaque. Discriminate between these because you have several readout options. I mean, the most traditional one is, of course, the plaques. So that means you have a confluent monolayer of cells. This is what you see in the far left. And all the cells that are still alive are stained with these kind of stainings. And the dead cells are not stained. So these are the white spots. So this is quite easy because you can count plaques often with naked eye, so you don't need a specialized microscope or any other reader to do that. You can scan these kind of plates, and there are several kind of plaque counting softwares, most of them also for free, but nevertheless it's pretty tedious because you need a good scanner, and you know, scanners are not made for scanning plates, but they're made for scanning paper. So it's sometimes a little bit tinkering around to optimize these kind of assays. Also, because you count often with naked eye, that means you need kind of bigger wells. I mean, nearly no one is doing plaque assays in 96 wells. So the minimum you have to start is 48 wells. A lot of people even do it in 6 well plates, but then this automatically means you need a lot of serum. For some studies, it doesn't matter, then you're fine. But when you have only a small amount of serum, especially when you have precious serum samples from animals, from wild animals, or also from children where you cannot get so much sample, then a plaque assay in a 6-well plate is not that favorable. There are also other plaque stainings where you have the reverse staining, where you actually can stain the infected cells. This is what you can see on the far right. So there we don't stain the living cells, we stain actually infected cells with a virus-specific antibody. This is pretty nice. because it's virus specific. So you are independent. If your cells die from something else, they will not be stained. And it's a little bit more specific because with this assay, you even could measure only one cell infected. So if you have a microscope, or I use an ELI spot reader for this kind of readout that can really distinguish a single cell from the background of non-stained cells, then you can perform a single round of infection neutralization assay. Nevertheless, I also wait for at least 2 or 3 infection rounds, because then, first of all, the foci are a little bit nicer, a little bit bigger. And I avoid kind of artifact stainings of single cells, so these kind of things. So this is why I also rely on multiple rounds of infection. You don't have to rely on stained foci with, in my case, the horseradish peroxidase. You also can have fluorescent foci. This you can either do when you replace your antibody with fluorophore conjugated antibody, or if you even have a recombinant virus that has a fluorescence tag. So then it's kind of the easiest, because then your virus itself glows already. And then, of course, infected cells will glow as well. For these, you then need also a specific microscope or a specific reader. So you see there are quite a lot of adaptations that you can do regarding your neutralization assay of interest. I want to shortly emphasize some benefits and disadvantages of certain neutralization assay formats. So first of all, the most traditional is the plaque reduction neutralization test. As I told you before, it's relying on plaques. So you have to have a virus that kills the cells that you're using. So this is kind of the biggest hurdle you have to overcome to establish such a test. However, it's really easy to do if you have the good cells and the good virus in hand, because staining of live cells is very easy, it's very fast, and it's cheap. And then it's kind of an easy and also very visual detection of the plaques by naked eye. So it's always really impressive. You can even, because you inactivate the virus, you can even show your plates to colleagues easily. It's a very visual test. However, the virus you use also have to not only have to kill the cells, so you need the CPE, but you also want to have regular plaques. But you can see already in the picture that not all plaques have the same size. So that's already not a real red flag, but you have to be careful that what you measure is real virus-induced CPE, and not that the cells die simply because of overgrowing. You sometimes see a ring at the edge of your well, then this also means that the cells overgrow and are not happy, and then they die, but not because of virus infection, simply because of stress. And also another disadvantage is if you have, for example, like these, the 6-well plates, then you count by naked eye, which can be very, very tedious if you have a lot of samples to test. This is why a lot of people prefer the FRNT, so the foci reduction neutralization test. For these, we often have a more or less automated readout. So what we do here is the staining of infected cells. So that means it's virus specific, as I mentioned before. And if you have the option, you can do an automated readout. This often happens with a plate reader, like an ELI spot reader. Some fluorescence microscopes come with a software that can do it. Some live cell imagers also can do plaque or foci counting. So there are quite some options. It really depends what is available in each lab. The other thing is that is really handy for the FRNT, you can downscale to much, much smaller wells. So for the foci assay, what you can see here is a 96 well plate, so that's very, very nice. It also allows you more replicates. So here I have triplicate testing, which is also makes the assay simply a little bit more stable. I heard that you can even more downscale to smaller wells than 96 wells if you have fluorescence based FRNTs. But of course, there are also some disadvantages. So you definitely need a virus-specific antibody. You don't need the CPE, but you need the antibody that is specific to your virus. And then you might need quite a lot, which might make the assay a little bit more expensive, because if you don't produce these antibodies by yourself, you have to buy them. And these assays are definitely not really made for naked eye analysis. So either it's very cumbersome to do it, or you really need a reader to do the counting of the foci for you. So when you have your foci or plaque counts, what do you do then with them? Of course, you simply can plot them on the left. You see the foci counts for each serial serum dilution. This is an example for 3 different viruses. And what you can see, the Zika, so the very light blue, virus induced no real neutralization, because you see that even with the lowest serum dilution of only 20, so 1 to 20 serial dilution, you reach nearly the same foci count than in the negative control. So that you can already see without any calculation that there's no neutralization. However, for dengue 1 and dengue 2, you see 2 different profiles for the dengue 2. So the middle blue one, you see that the neutralization is really strong, because in the first 1, 2, 3, 4, 5 dilutions, you have no foci at all. So that means there's no infection for these serum dilutions. So this translates into that they are neutralizing antibodies at a very high concentration. However, for dengue 1, so the dark blue one, you see that you have only a very small amount of infection events in the lowest serum dilution, so at 20, and then it's constantly rising. So that means there are neutralizing antibodies, but they are not a lot. So what do we do then? A lot of people work with the 50% of reduction. So you always have your negative control, which is, and you can even compose your negative control out of several components. So what you always should use as a negative control is the virus only, so nothing added. And even better is when you then also have the negative control serum, so there's no neutralizing antibodies in it, and then you add them in the same manner like you use for your serum that you want to test. And these 2 negative controls should come to the same plaque or foci count. In this case, it's 60. So, and then you say, okay, and I determine the titer of neutralizing antibodies where I have 50% reduction of my negative control, which would be in this example, 30 foci. And then you check, okay, where do you have a 50% reduction? So where is the 50% reduction threshold crossed by your data line? If you want to be much more specific, especially in the background of cross-reacting viruses, we often set a much lower threshold, which means a higher restriction, by looking for 90% of reduction. So when we go back to the 50% reduction, what we then measure is, for this example, a FRNT50 titer. So that means we look for the reciprocal serum dilution that induces 50% reduction of foci compared to your controls. So as I mentioned before, Zika has no neutralizing antibodies, because you see the curve doesn't cross the 50% reduction line. And for dengue 1, the FRNT50 titer is much lower than for dengue 2. However, you also can calculate these titers, which is easier than always looking for a line and where is it crossing. A lot of people use a prograde regression analysis as a binary response model for the calculation of neutralizing antibodies. So you simply use the statistics program of your choice, and then you enter your data. And what you can see that for dengue 2, the curve is fitting very well, whereas for dengue 1, the slope is not really matching. However, I have really good experience with the log prograde regression, so this is my favorite method. And you see also that the calculated FRNT50 titers, so these are in the gray circles, are not that different from what we saw by simply observing the 50% reduction. And of course, you can do the same for any FRNT or PRNT titer you want. There was a very interesting publication quite a while ago where Hendrik Salje investigated which reduction line would be the best. So is really the FRNT50 or 90 the best? He actually found out that for dengue, PRNT75 is the optimum evaluation point to say, okay, this is the threshold of neutralizing antibodies. And interestingly, he also found that the choice of the statistical model is not that significant for the calculation of the relative titers. So feel free to go further into it if you are a statistics nerd. Now I want to move further into the application. So a lot of people are interested in arboviruses. There are quite a lot of them for humans and also for public health. The most interesting ones are, of course, the Flavi and the Toga viridae. So the flaviviruses and the alpha viruses. These are also quite okay to work with. There are more and more people looking also into the Bunya viruses. They are much harder to work with because they grow really slow. They are harder to isolate because they have a segmented genome. They also tend to make heterologous recombination events. So they are much harder to work with. So I will focus on Flavi and alpha viruses. These are the viruses that we also work with in. So arbovirus diagnostic is often a little bit challenging because you have several steps you can diagnose an arbovirus infection. You can look only for the clinical symptoms, but this is quite tricky because a lot of arboviruses present with the same symptoms. So a lot of people have flu-like symptoms, they have fever, but it's pretty unspecific, especially in the tropics. So, but what you also can do, of course, is a molecular diagnostic. It's super specific. You definitely or you directly measure if there's virus present. So it's the cleanest way of diagnosing a viral disease. However, for arboviruses, the diagnostic window is often pretty short. A lot of people have a viremia only around 1 week. Of course, you have the PCRs are really sensitive, so you might even extend this window, but nevertheless, if the viremia is over, if the virus cannot be found in the body anymore, you cannot do molecular diagnostic. So especially if you want to investigate huge cohorts of non-symptomatic or simply a cross-sectional study, and you want to see, okay, what is the prevalence of this virus in the general population, then the likelihood that a person is acute infected and has acute viremia is very low. So you're depending on serological diagnostic. This is pretty handy because antibodies persist for years. You can even measure 2 kinds of antibodies, so IgM and IgG. IgM normally indicates that there was a recent infection around about 3 to 6 months ago. But the problem with serological diagnostic is it's prone for cross-reactivity, because especially for flavioviruses, because they're antigenically very homologous. So what are we doing with the arbovirus NT at IPC? We have quite a zoo of viruses where we can perform NTs. It's not restricted to arboviruses alone. We, for example, also do COVID neutralization assays. So as long as you have your virus in hand and you have a good run cell and virus culture, you can do quite a lot with this kind of pipeline. At this point, I really want to emphasize you need a really strong team for performing and having an NT pipeline like we have. You need great people in the field to get you all the interesting samples. You need a huge lab team, a well-trained lab team. You need, of course, the contribution of the communities, that they let you sample, for example, here their dogs to check on them. So it's really a team effort to have a well-run NT platform like we have here. So I'm very happy that I can present you our data. For example, one very early study that I did here in Cambodia was a chikungunya neutralization assay. We had an outbreak several years ago, and then afterwards we wanted to investigate why was there such a discrepancy between people who were born before 1975 and after. So there was quite a suspicion that chikungunya was circulating in the 70s already in the population, and this is why these people kind of had a pre-existing herd immunity. So we did NT with different genotypes of chikungunya. So not only serotypes, even it's really comes down to genotypes that we could see. And when you look on the left, you see in the IgM negative people, so the people that were not affected by the recent outbreak, you have a broad, already a broad neutralizing antibody response. So this really indicates that this older population was already exposed to Asian genotype virus. If you want to read further into it, you can check out the publication. One was the first investigation of the Madeira Island outbreak from 2012. So that's in Spain. We had a collaboration partner there, and they were looking for people who can do a dengue neutralization assay for sero and genotypes. So we did this, and with that one, we really could confirm, yes, it was a dengue 1 outbreak, and we even went further and said, yes, we can also confirm that it's a dengue 1 genotype 5 outbreak. This was very interesting because certain genomic analysis already pointed out there was a high exchange of people to South America. So we would confirm, we could confirm this with a serological analysis. That was quite cool. What I like to spend my last minutes on is some Japanese encephalitis seroprevalence studies that we did in domestic animals. So we had several studies where we were collecting samples from domestic birds, but also pigs. And due to rabies vaccination campaigns, we also had samples from dogs available. So what we did was simply running neutralization assays against Japanese encephalitis and dengue. In this case, it's dengue-free. And normally, I like to point out, normally birds are not known to get infected with dengue. So the neutralizing antibody response in birds should be 0. But what we saw with the neutralization test, it's actually quite similar, which is not the biological reason. So there could be either, okay, no one ever looked in birds, so maybe they really get infected with dengue. But from our investigation, we think it's more likely that these birds get infected with Japanese encephalitis. And then simply the antibodies they develop against Japanese encephalitis are cross-reacting to dengue. So that means we cannot distinguish between Japanese encephalitis and dengue neutralizing because the antibody response that these birds build is too similar to each other. We also had the samples from the dogs and the pigs, and there the picture is much, much clearer. Yes, we also saw a neutralization response against dengue. However, the responses were significantly different. So we can say in the same sample, the neutralization response against Japanese encephalitis is significantly higher than against dengue. So our assumption is then these animals were infected with Japanese encephalitis for sure. So that points out that pigs and dogs might be very good sentinel animals for Japanese encephalitis. However, there's a problem. Pigs have a high prevalence for Japanese encephalitis in Cambodia, for example. So when they reach the age of 6 to 9 months, nearly all of them are infected. So, and if they're already seroprevalent, then they are not a good sentinel animal anymore. Another problem. Yes, there was a significant difference between dengue and Japanese encephalitis, but what is with the viruses that are within the Japanese encephalitis serocomplex? So the other viruses like Usutu virus and West Nile virus, they are very close related to Japanese encephalitis, and therefore the antibody response can be overlapping. And this is exactly what we see in the pigs. So you see most of them, yes, they had Japanese encephalitis virus antibodies, but these antibodies were also reacting against West Nile and Usutu. And for these samples, I then cannot say, okay, yes, this pig was infected with Japanese encephalitis. It could be any virus from the Japanese encephalitis serocomplex. However, for the dogs, yes, there was also over 1/3 of the dogs also showed this cross-reactivity. However, there were 12% of the dogs that showed a single reaction to one virus. That was really nice. So I could really discriminate between dogs that showed antibodies against Japanese encephalitis or only against dengue-free or only against West Nile. So that was really nice. So this points out that dogs might be actually a nicer sentinel animal species. This comes also in handy because a lot of One Health initiatives, especially for rabies, they use dogs. So they vaccinate dogs against rabies, so dogs can be handled easier. Also, dogs can be handled easier in terms of sample taking, because farmers often don't like when you sample their pigs. They get stressed, they lose weight. Farmers hate that, but a lot of people don't mind if you vaccinate your dog and then also take some blood from them. So with this, I'd like to finish and point out that dogs might be a very important a part of serological surveillance because they can serve as valuable sentinel animals for arboviruses. All these studies that we are doing are funded by a broad range of organizations. I really like to thank them because these essays involve a lot of people, and I'm very thankful to work with them. And with that, I'd like to thank you for your attention.
Evidence of crossing reactions of virus in pigs, birds, and also dogs and mammals. So if you have any questions, please send us a message through chat box. Or you can raise your hand in the WebEx.
I don't think there are any questions in the audience, and anyways, we would have to relay them to Heidi, who is not with us. But just to give a bit of perspective, this is exactly the kind of techniques that we want to use in the next phase of the CDOC project that we mentioned. The idea is to explore the prevalence patterns of dogs and their dog owners to try to assess the role of sentinel for 3 arboviruses, dengue, West Nile, and Japanese encephalitis. And we are currently putting together more proposals to secure more funding to not only do the survey in Indonesia, Thailand, and Cambodia, but also provide some training on the specific techniques that Heidi has very thoroughly presented before. And I think we may need to move to the next speech, which would be the last of the dog session by Prof. Johan.
Okay, so thank you, Dr. Michel. So the next speaker will be Dr. Johan Michel. So he's from University of Liège, Belgium. He's gonna present the topics is interest of microbiota, analysis of wild and domestic species for the study of pathogen transmissions. Please.
Okay, thank you very much. Can you hear me and can you see my screen?
Yes, we hear.
Okay, that's perfect. So yes, thank you very much for the invitation. It's always a pleasure to meet you, to meet all of you. And I just regret that I am in Belgium during the winter and that you are in Thailand with a much more better weather. But anyway, it will be for the next time. Yes, I will present you today, yes, different informations, but unfortunately concerning the project on dogs with the COVID pandemic, we have lost many times. And so for the moment, we do not have much results concerning this project. So I will more present you a more general information about the interest of the microbiome studies, etc. And I will present you the aim of our project concerning the dogs, but also concerning other species that we are studying in Indonesia. So first of all, if the PowerPoint is working, yes. First of all, I will remind you some general concepts. And the first concept that I would like to remind you is the holobiont concept. which is precisely the view that animals would not be autonomous entities, but rather they would correspond to holombionts, which comprise the host, but also together with its associated microbes. So, you know, we will be a kind of a whole entity corresponding to the host and to the microbes. So these microbes interact with their host in many ways, and they can have a pathogenic, mutualistic, or commensal role. So this is the interest to study all these species in a whole entity. And so the field of microbiome research can shed a new light on many biological processes, from the human and animal medicine, for example, but also for a better understanding of the ecosystem functioning, and also for the conservation of endangered species that we are studying in more particularly in my laboratory. And so the microbiota is defined as the entire community of microbes which are present in a specific body part of a living host. And so it comprises notably the bacteria, the archaea, the viruses, the protozoa, and the fungi, for example. But in our context, we are more focusing our researches on bacteria, but it can be also done on other kind of microbes. And so, with the extensive development of molecular techniques like the next generation sequencing methods in the last 20 years, it is now possible to obtain very, very precise information of a large part of this microbiota. And therefore, it's very interesting because with all this information, we can more precisely explore the intricate relationships between the host and the microbes. And so to develop such kind of studies, of course, you need to have different material, biological material. So you can go in the fields, for example, as we did in the past with Dr. Sachmoran in Thailand, for example. So you can collect different rodent species, for example, and so you will be able to collect different tissues or organs from this material. And so each of these organs will be characterized by a particular microbiota. So it is interesting always to study different kind of organs if you want to have a whole idea of all these microbiomes which are present in these animals. But also more and more, it is interesting to use non-invasive material. And so it is particularly very interesting if you want to study, for example, endangered species, which are very, very elusive, which are very rare. And so, for example, you can collect feces in the fields. And so you will have interesting information about the intestinal microbiomes, for example. But you can also, for example, here for the dogs, collect saliva material, and so you will have a good idea about the microbiomes which are in the mouth of the animals. And of course, it is a much less aggressive method to obtain material. And so as soon as you will have this information, so I will not explain, I don't have the time to explain you all the processes of the next generation sequencing, but so after the next generation sequencing, you will obtain millions of sequences which correspond to hundreds of here, for example, bacteria strains. And so the first data visualization, very general visualization, correspond to a pie chart, which will give you the most important strain, the most important groups of bacteria which are present in your samples. But you can also use, for example, heat maps, which are very interesting because you can have very quickly a general idea of the presence of some bacteria or family of bacteria in one sample. So, for example, here each linear, vertical line corresponds to one sample, to one individual. And so more you have a red color, more you will have the presence of one bacteria strain. strains, which is represented here on the right. And if you have a black signal, this means that you don't have in these animals, in these individuals, the presence of some of these bacteria strains. So it is quite interesting to have a very general information about the microbiome in your sampling. So I will give you some examples. So as we don't have yet many results for the CDOC project, I will just show you 2 examples which were obtained in other regions of the world. But after I will summarize the project that we have in Southeast Asia. So the first example corresponds to the study that we developed several years ago about the impact of the climate change on the emerging of the Lyme disease in Canada. So it's quite far from Southeast Asia, but anyway, we could very easily translate such kind of study in Thailand, Cambodia, or Indonesia, for example. And so the system of this Lyme disease in Canada is represented by these 3 species. So we have one reservoir, which is a small mouse, Paromyscus leucopus. We have a vector, which is a tick species. Here it is Ixodes scapularis. And we have one pathogen, which is one bacteria, Borrelia burgdorferi. And the problem is that as the winter are less and less cold, we have year after year a better survival of the reservoir and of the vector in many regions of Canada. And so, of course, if you have an increasing of the number of reservoir and vector, there is much chances that you will have also the disease which will appear. And it seems that it is the case as we have more and more Lyme disease cases in Canada. And so the aim of our study was to develop a kind of monitoring of the presence of the Lyme study, but also the presence of the Borrelia bacteria in the reservoir and in the vectors in many regions of the French-speaking province of Quebec in Canada. And so to obtain this information, we developed bacteria metabarcoding. So we perform next generation sequencing method, which is based on the sequencing of 2 hypervariable regions of the 16S RNA gene, which is the most common gene to study the diversity of the bacteria. And again, we obtain this kind of heat map, which represents a summary of all the bacteria strains that we obtained. And the first interesting results that we had is that we were able to evidence the Borrelia bacteria in many regions of this Canadian province, which is called the Quebec, with a sensibility of detection which was at least 4 times higher as compared to the classical PCR. So we were very surprised to obtain a much more precise information about the presence of this Borrelia. But the interest also of this microbiome approach and next generation sequencing approaches was that we were also able to evidence different co-infections in many hosts among different pathogens. And so in many hosts, for example, we evidenced the presence of Borrelia, but also the presence of other bacteria strains like Bartonella, like Ehrlichia, which can also give important sickness on human populations. And so following these approaches, we were able to evidence that often people, when they are bitten by ticks, they can also suffer of co-infections, which give much more stronger sickness in these regions. And definitively, according to these informations, we evidenced a link between warmer winter and the better survival of the host. So these mice, Paromyscus leucopus, the vectors, the ticks, and of the Borrelia bacteria. And so, according to this information, we are afraid that the Lyme disease will continue to spread in Canada in the future, in link with the continuing importance of the climate change. And so another example was the study of the links between the microbiome and the immune system, but also the environment. And so according to the different studies which are performed on the human, it seems that a good microbiome diversity would help to maintain a good health. And so our questions working on the wild animals was, is the microbiome directly linked to the environmental factors, for example, the diet that the wild animals will eat, but also to the habitat where these wild animals are living. Another question also was, is the microbiome linked to the immune system of a host? Again, according to the information that we have for the humans, it seems that a better immune system, a good diversity of the immune system would help to avoid overdominance of some bacteria strains, which could be a problem for the health of the animals. And also it would help to avoid the presence of pathogens. And so to have a better idea about this concept, we worked on one critically endangered species, which is the case of the European mink, a small Mustelidae species, which is almost extinct and which is only surviving in some European areas. And so we worked on the microbiomes, but also on the immune system genes of this species, more particularly the the major histocompatibility complex genes, so it is always difficult to say. And the first results that we obtained was that the microbiodiversity of these animals was directly linked to the diet of the animals and to the environment where the animals are living. And so we evidence very different microbiomes according to the habitats where these animals were living. And more particularly, we evidenced also that the microbiome diversity was directly linked to the quality and the diversity of the food. And so such information was very important and interesting, as this species is so rare that we are performing conservation breeding for this species. And so often in these centers, we give food which is not really natural, and it has a deep impact on the diversity of the microbiome and then on the health of the animals, and particularly if these animals are released in the wild. We also evidenced that the immune system genes, so again, the major histocompatibility genes, do not systematically help a diversified microbiome, but rather it seems that these genes helps to select important bacteria strains, which are very interesting for the survival of the host. So we also obtained many other results, but I don't have the time to explain you today. And so to come back to Southeast Asia, so as I explained you, we have at least 3 different projects for the moment, but we have many problems to obtain results because of the COVID pandemic, which do not allow the mobility of researchers from Europe to Asia and from Asia to Europe. But anyway, I will summarize very quickly all this project. So the first project that Michel probably already explained you is the case of the rabies and dogs in Indonesia. So this is the CDOXY project that Michel already presented you. And so this project is very interesting because it is a multilateral project which associates Indonesian colleagues, French colleagues, and Belgium, myself, colleague. And so the main aim of this project is multilateral. So it has as aim to perform socio-ecological studies in different Indonesian areas, using, of course, the One Health approaches. So again, to consider everything as a whole. The other aim is to study the rabies virus, notably, and its circulation among the dogs and the owners, the human owners, and also the aim, which will be more focused for myself, it will be the study of different bacteria communities using next generation sequencing technologies. And so this project is developed in collaboration with Professor Wayan Artama, notably from the UGM in Indonesia, Dr. Michel Degarin, that you know, Dr. Serge Laurent, that you probably also very know, and myself. And so the aim of the microbiome aspect will be to monitor the zoonotic pathogens in dog and to have a better idea about the shared microbiomes among the dogs, but also between the dogs and the owners. So the work is in progress. The material, a great part at least of the material is already collected in several Indonesian and Thai areas by our Asian colleagues, and particularly the Indonesian colleagues. And so we hope that the microbiome analysis will be performed in 2022. And so again, these researches are in standby for the moment because of the COVID pandemic. So the second project that we want to finalize is the case of the Sumatran elephant. And more particularly, we would like to have a better idea about the circulation of different pathogens, and particularly, for example, the tuberculosis bacteria among the wild and captive elephants, which are living in the southern part of Sumatra, but also the circulation between these elephants, the cattle, the bovine, for example, and also the human population living in this area. And so again, we aim to use a microbiome analysis, and this analysis will be focused on faces, which will be and which are already collected on these three groups of hosts. And so this project is developed in collaboration with the professor Vishnu Nurkayo at the UGM in Indonesia, and Michel de Garin again, and myself. And again, this research is in standby for the moment because of the COVID pandemic, but we hopefully that this project will be able to continue in the next year. And the last project concerned the managing of wildlife, livestock interface. And the aim of this project is to better understand the conservation and the health of the Rusa deer in Java and West Bali, which is a species which is endangered in some Indonesian area. And so more precisely, we aim to better understand the interactions about the deer, the livestock, and the human. So as for the elephant project, and because we hypothesize that this interaction will increase the probability of disease transfer, and also which will increase the competition for water and forage resources, which often lead to conflict with farmers and these wild species. And so the aim of our microbiome project will be to screen the parasites and the pathogens in deers, but also in the livestock populations, which are living in surrounding areas near the protected areas where these deers are living. And again, we will develop a microbiome and meta-recording approaches. So this project is developed by the collaboration with the Dr. Subrata, Sena Subrata, and Professor Vishnu Nurkayo from, again, the UGM, but also the Professor Wayan Artama, Dr. Michel de Garin, Serge Morand, myself, again, all the same people, as we are good friends and we like to collaborate all together. And again, we hope that this project will be able to continue in next year. For the moment, at least 200 faeces samples coming from different protected area on the eastern part of the Java island are already collected, and the DNA is already extracted. And so we hope that Dr. Sena will be able to come in my laboratory at the beginning of 2020 to perform this analysis. But again, we are in standby again due to the COVID pandemic. And so in conclusion of this talk, we can say that this microbiome and this microbiota concept, as well as the development of these new technologies like the next generation sequencing methods, are revolutionizing our knowledge concerning the relationships about the different microbes and pathogens and their host. And so our hope is that we will be able to develop all these concepts in Southeast Asia. But again, we are waiting to have a better situation concerning the COVID pandemic. So I thank you very much for your attention. Thank you.
Dr. Johan. So right now, is there any question from the floor? You can ask by yourself, or you can send me a message. Then I will deliver to the speaker.
There are two questions in the chat. There is one from Dr. Pizzano. You want to ask the question live to Dr. Johan? Okay, if you're not online, maybe I can relay the question. So Johan, Dr. Pizzano was asking, how do you interpret the data from the new generation sequencer? Is it based on existing databases, data bank?
Yes, mostly it is based on the data bank. Yes, we can identify quite precisely, but at least at the genus level, but also at the species level, the bacteria strain that we obtain. And fortunately, we have a good database for the moment in the public databases for the bacteria. And year after year, these databases are improved. Yes, but
And we had more precise questions asking how much each does cost and if he can send samples to you. So I will leave that to you, maybe for direct interaction.
We can talk. Yeah, we can talk on the side. It depends of the number of samples, of course, because when we perform run, we can analyze at least 200, 300 samples at the same time. And so if you just send 10 samples, but so the cost will be different. But we can talk after about that. But it would be a pleasure, of course, to collaborate on this aspect.
Thank you. There was another question by Dr. Nicolas. Nicolas, are you online or do you want me to relay the question? Okay. So Dr. Nicolas Lenné was asking, he said it was a very interesting approach, especially considering that for human, elephant, and bovines sharing zoonotic diseases. Do you directly work with mahouts and or farmers, referring to the project you mentioned on the Sumatran elephant?
Yes, we have the chance to collaborate with Dr. Vishnu Nurkayo, who have a very good contact with mahout, but also Dr. Harry, Professor Harry, who is present also in this conference, and so and also a professor of the University of Lampung in Sumatra. So we have the chance to have all this collaborator who know the mahout and all this project on the elephant, and so the aim is to collect feces from the captive animals and also blood samples. Dr. Harry already collected all these samples, and we had a common PhD thesis with a student. And the most problematic concerned the wild elephants, because it's quite difficult to collect feces of wild elephants. So we tried, and so it's not easy. And because of the COVID pandemic, we were not able to continue the research of these feces. But our aim is to come back in the fields and to go, for example, near the ponds during the dry season, where the elephants are more attracted, and the idea is to collect the feces, for example, around the ponds. It's just one possibility. But for the captive elephant from the cattle, it's not a problem, as we already have good collaboration with our Indonesian colleagues.
I think we are running a bit behind time, so and it may be getting to the time where we just need to wrap up the dog session. So I hope that you're all convinced about the diversity of topics and the diversity of approaches that we are trying to merge to have this social, ecological, and genetics associating very different approaches, but to make sure that we understand all this interaction that the wildlife, livestock, human interfaces and disease transmission in such specific areas. So I would like to thank very much all the presenters for the dog session, because it's been very diverse, very, very live. And I think that it may be time now for moving to the next session in this afternoon.
Thank you very much. Thank you, Michel.
is Professor Dr. Chen Cheng-Wen from Department of Biological Research, National Chiayi University, Taiwan. He's gonna talk about the population genetic structure of strip snakehead and their parasite in Taiwan and Thailand.
Good morning, everyone. And this is Xianwen Chen from National Chiayi University, Taiwan. It's my honor today to be in this meeting and share some of my recent researches with you. My topic today is population genetic structures of striped snakeheads, Shonos striata, and their parasites in Taiwan and in Thailand. First, why should we concern invasive alien species? As we see in these pictures, 86 of islands' animal extinctions are related with alien species. So alien species present a major threat to the local biodiversity. Also, when we introduce an alien species, we not introduce the species itself, but also some parasites and pathogens with them. For example, in this picture, Tilapia is one of the most widely introduced aquaculture species all over the world. So the Tilapia originate from East Africa. In the East Africa, they carry 23 different species of parasites. But when they introduce a new places, usually their parasite diversity will reduce. And sometimes they will bring their parasites with them. For example, when Tilapia introduced to Japan, they bring 3 species of parasites to Japan. And sometimes they didn't. For example, when the Tilapia introduced to Taiwan, they didn't bring any parasites. But when they arrived Taiwan, they actually pick up 2 parasites from local fish. So the alien species will change the host-parasite interaction in the introduced area. So they may change this interaction by, for example, co-introduction with their parasites. And these co-introduced parasites, sometimes they can also infect the local fish. And we call this spillover. And even though they didn't bring their parasites when these alien fish were introduced, they may get the parasites from local fish. We call this spillback. And if the spillback parasite didn't grow very well, and it is possible they can dilute the population density of a local fish, the parasite population density of the local fish. So we call this dilution effect. So indeed, the alien introduce an alien species can change the local host parasite interactions. And today we will look at this specific fish called striped snakehead, Shonos striata. Shonos striata is native to South Asia and Southeast Asia. And as a common aquaculture species, they were introduced to the east part of Indonesia, the Philippines, and in Taiwan. So when they were introduced to Taiwan, which is around 50 years ago, in 1970s, They were used as aquaculture species, but soon after they were introduced, they will escape from the aquaculture pond and find in the water body outside the aquaculture pond. So first, they were caught in wild in 1983. And because this fish is voracious, they are voracious predators. They eat other fish, they eat small turtles, they eat Gemini birds, they eat almost everything in the water body. So now they become one of the dominant and most commonly found fish in the freshwater ecosystem in Taiwan. You can find them in dams, the ponds, lakes, the rivers, and the paddies. And basically, they are everywhere. So in order to understand how these fish, their invasion histories, and the potential, the parasites risk of these fish bring with them, we study Striatas from 8 different locations in Taiwan and 2 locations in Thailand. So first, we're using the mitochondria cytochrome B genes as the marker to study the phylogenetic history or phylogeographies of this introduced Striata fish. Using cytochrome B, we find 9 different haplotypes, which belong to 2 different lineages. The lineage 1 are samples from Taiwan and Thailand. The lineage 2 are samples from Thailand, in particularly in northeastern Thailand, the Udon Thani area. So when we compare the sequence, we find the lineage 1 is matched with the clade 4, which mentioned in Ruber's 2020 paper. And this clade 4 fish is widely distributed in Indochina and in Sunderland. The clade 2, which we find in Udon Thani, is matched with the clade 1 fish, which only find in Indochina area. So these 9 different haplotypes, their distributions. In Taiwan, we examined or we sequenced 103 fish. We find only 3 haplotypes. Only 3 haplotypes. So this hepatitis 6 is one of the most common hepatitis, which is also found in Thailand in Nan area. So in Thailand, we sequenced 17 fish. We find 7 different hepatitis. Particularly in Udon Thani, 12 fish has 6 different hepatitis, including the clade 1 and clade 4 type. So if we look at the pairwise gene distance for the 8 different populations, local populations in Taiwan, as we mentioned, we find only 3 different haplotypes. So the gene divergence is very low between different populations. The only exception is the Maori population, which seems to be a little bit different compared to all other local populations. We only conduct the molecular variation analysis for Striatas. We find between different countries, different populations, and within populations, their variation are all significant. Finally, we conduct mismatch distribution analysis using these cytochrome B sequences. In Taiwan's population, we find it presents a unimodal distribution and implied a potential single introduction and the following the rapid expansions. In Thailand, we find these multiple or bimodal distributions and implies potential multiple introductions or mixed populations. So the first part, we can make this small remark. Regarding the population structure of Shonos striata in Taiwan and Thailand, we find the introduction of Shonos striata to Taiwan is probably a single event, or it may be a multiple event, but from a single source. So this source is supposed to be close related to the clayfall fish mentioned in Ruber's paper. And this clade is widely distributed across Indochina and Sunderland, and probably the one who used mostly in the aquaculture purposes. And the introduction of Shonos striata in Taiwan has experienced the rapid population expansion without too much population isolation, which also indicate a potential human transfers or human transfer of this fish among different rivers. Now let's look at the parasites. In Thailand, we dissect 18 fish and find 3 species of parasites, including 2 Encephalitis and 1 nematodes. In Taiwan, we dissect more than 400 fish and find 8 different parasites, including 2 Encephalitis, 5 nematodes, 1 trematode. So if we compare parasites from the native reach, for example, in Thailand, and those in Taiwan, we find 2 parasites were shared between the native range and the invasive introduced range. They are Encephalon, Pericentis species, and also Camellias species. These 2 species, Pericentis species and Camellia species, also present 2 of the parasites with highest prevalences and the intensities in all the locations we surveyed. In addition to survey the Shonos striata, we also examined other fishes within the same pond with the Shonos striata in Taiwan. In this part, we examined more than 400 fish, including 3 local fish, native fish in Taiwan, and 7 alien fish. In this fish, we find only the alien fish were infected with the parasites. 2 alien fish infected with parasite. And again, Pericentis, these are Encephalitis, infects the auxiliaries and also Trichopodes, these 2 alien fish. So let's look at the morphology of Pericentis, these are Encephalitis. In this species, we find they are actually distinct from other Pericentis species in their proboscis hook, their proboscis receptor, and also giant nuclei number and the body length. So if, in addition to morphology, we also sequence this parasite, this Encephalitis. We're using the ITS gene to look at the differences. So this is the samples we got from Shonos striata. And this is other Pericentis species. So the Pericentis species we study from monophyletic groups. However, they separate to different lineages. And these 2 lineages actually represented those samples from Udon Thani in Thailand, and samples mostly from Taiwan and river in Thailand. So if we look at the genetic distance from the Pericentis species, we collect from Striata and other known Pericentis species, their distance is more than 10%. But if we use the mitochondria COL1 gene to look at these specimens, this time we find 3 different lineages. The lineage 3 represented also those from Udon Thani in Thailand. The lineage 2 are presented the samples from Taiwan. Lineage 1 presented the parasite samples from Taiwan and Nan River in Thailand. Also, we find this Trichopodus host. Their Pericentis species also were grouped into this lineage 1. If we look at this distribution of haplotype, we find total 16 haplotypes, including 7 haplotypes in Thailand and 9 haplotypes in Taiwan. So this is quite high diversities. Particularly in Thailand, the 7 parasites we sequences actually result 7 different haplotypes. So if we look at the pairwise distance, pairwise FST, and we find in Taiwan, 4 different local populations, they has a quite divergence from each other. So if we look at molecular variation analysis, we find most of the variation from between countries and within the populations. Among different populations, they actually didn't show the variations. We also conduct a mismatch distribution for the parasite Pericentis species. In this case, we conducted for 3 different lineages we find using the COL1 genes. The lineage 1 represented the samples from Taiwan and Thailand. We find it form a multimodal distribution, such as a mixed population with expansions. The lineage 2 show unimodal distribution and suggest single introductions. Lineage 3, which we'll find in Udon Thani, northeast Thailand, we find this show the bimodal distribution, also suggests a mixed population with expansions. The second shared parasite species, nematode Camellia species. So we compare the Camellia species we find in this study with other known Camellia species. We find the buccal size and shape. Also, the basal ring size is quite different. More importantly, so far, all the Camellia species will find this structure called tritons. This structure tritons in other Camellia species, but in the Camellia species in our study, we didn't find this specific structure. So this shows that our species is very different. We also do the molecular analysis. For example, we're using the COL1 to support our Camellia species form monophyletic groups. In contrast to all others Camellia species, including this common Camellia corti, which commonly find in the ornament fish. So this is the pairwise distance. of our Camellia species to other Camellia scotti population or other Camellia species. We see within our species, the variance is less than 1%, but in contrast to other species or other population, in coatis and in this hypothermicity, hypothalamicity species, actually all the difference from larger than 15%. In Taiwan, so far we only sequenced 18 parasites from 5 different populations. We find only 2 hepatitis. So the second remark here is for population structure of parasites in Striata. First, the Pericentis species and the Camellia species recover from the Striata in this study. Probably they were new species. So based on CO1 genes, the Encephalitis Pericentis species were divided into 3 different lineages and 16 haplotypes in Taiwan and Thailand. But for Camellia species, we didn't find too much genetic variations. And if we compare the host parasites phylogenies using the Pericentis species, we actually find strong host association. This is what we call the host parasite association. First, for the host species, Striata, this is the cytochrome B gene tree. So we find in this group, these big clades, which related to Ruber's clade 4, we'll find in many in Taiwan. And this clay collected from this fish collected from Udon Tane will associate or match with the clay 1 in Ruber's paper. They form actually quite nice with Pericentis species. This is using ITS genes. This is a lineage 1 and this is lineage 2. And one thing worth to notice is this. In this big group, we find 2 fish, 2 groups of fish from Thailand. For example, this group is from Udon Tane, but they are associated with clay 4. These groups from Nan River associated with clay 4 fish. So because they are associated with the clay 4 fish, their parasite were actually located in this lineage 1, not the lineage 2 in Udon Tane. So here you see, even though these fish are from the same location, but their parasite will form different phylogenetic lineages. So finally, I will use this picture to conclude my talk today. So when Striata invaded to Taiwan, we think they bring their own parasites. Here, mostly the Pericentis species and the Camellia species we find. So we see the co-introduction here. And particularly for the Pericentis species, it actually spill over to other fishes in the same pond. However, this other fish is not the local fish, but also an alien fish. So in the future, we hope to get more Striata and their parasite samples in Thailand and in other native locations in Malaysia and Indonesia. Also, we need to do more comparative works on morphology and molecular characteristics for these 2 commonly shared parasites from Striata. Finally, I'd like to urge more international collaborations between my university National Giant University and the Ventec Kasesa University. And hope after the pandemic, we will start the communications and the exchange. With that, I'd like to thank my graduate student, Mona Lee. She did the most of the work of these presentations, and my other members in the lab, also some professors, Professor Huang Dajun, Liang Shixiong, Zhen Qingxian, Yan Shenghong, Fan Jiakun, and Wang Hongyi. These are my local collaborators. And my international collaborators, Professor Ozos, Dr. Nita, Professor Tong, and Professor Serge, and also Professor Alexis. In particular, Serge and Tong in Thailand helped me a lot during our sampling this Triata in Thailand. My research is funded by new southbound talent programs by Ministry of Education and the Ministry of Science and Technologies in Taiwan. So welcome to the parasitologies, and thank you. We hope we will resume our Thailand parasitology summer camp after the pandemic. Thank you.
Okay, thank you, Professor Dr. Chen Sheng-wen. So, is there any question from the floor? You can send.
If not, I can have a question. First, I would like to thank Seven because we were together in another meeting. We just quit the meeting 2 minutes ago, actually. And we joined this meeting together, back to this meeting now. Thank you very much, Seven, for all of your work on this. And I hope you will continue with your students to give a lot of these very fascinating talk on using the genetics of this, especially the host. I have just one question. Do you think that because you have Thailand and you have Taiwan, do you think you need to expand a little bit more in other countries of Southeast Asia you study?
Yes, indeed. I think you just missed my last slide I mentioned. I mentioned like we do like to have more collaborations, like for example, like in Malaysia or Indonesia, because the fish, these Shana striatas, they're widely distributed, and the clay we are looking for actually commons in Malaysia and Indonesia. Also, is a very common aquaculture species. In addition to this specific clay of fish and their parasite we are looking at, actually, I think this will be a very nice example to look at the host parasite co-evolutions in Indochina. So in Indochina, we have several different clades of this Shana striata fish. So I expect we will find actually there will be nicely co-evolutionary clades of the parasite as well for the Encephalitis pericentis. Yes.
Thank you very much.
Thank you. Thank you for your questions.
Okay. Is there any question? Okay, thank you. So we will go on to the next speaker. For the next speaker is our Assistant Professor Dr. Alexis Libas from University of Barcelona, Spain. So he's gonna talk about helminths in wildlife.
Hello, good afternoon. Okay, I will about my presentation. I will speak about the helminths in wildlife, and in the case of rodents. Why? Because we have a long tradition in my laboratory of parasitology. I will make a small introduction about this story, and after I will explain the more recent studies that is finished short time ago, or that they are in the way to be finished. In our case, we choose the rodents like a model of a study because it's a very diverse group, and mainly in the family Muridae is where we work more. In this case, the family Muridae is very well represented, and concretely the subfamily Murinae, that is the one with more species, is the one that we work more intensively, obviously, because it's the one that it's more a species. A little bit the story of the why we start to decide to study the parasites of rodents. We started in the 70s in my laboratory with a thesis that was first in the 70s. I started to study the small mammals in Catalonia, the helminths. After a pass to the helminths of the small mammals in Spain, Iberian Peninsula, and after we moved to the Balearic Island in the 80s to continue with these studies. After that, also we continue increasing in the years, like 85, the systematic study of all the rodents in Spain, and finally we increase this study to also to carnivores or lagomorphs, but I will not speak about that. And more in the 2000s, we start to increase the surveillance of helminths of rodents to the Atlantic islands of Canary Islands, Madeira, Azores, and Cape Verde, and also in the Western Mediterranean islands like Corsica, Sicily, Sardinia. This can be very clear. See what is this process. For example, what I show here is the Catalonia Biodiversity Catalogue. In this case, I just finished to prepare the trematodes. We have to provide a list of all the trematodes of parasitic trematodes of Catalonia, and as has been done for many groups. Here you have like an example. And what is interesting is 42% the trematodes that I was able to record in the literature review. Of the 80, 42% it belongs to small mammals. It is clear that this is completely biased because our team has worked a lot in this topic for many years. Of course, this is something irreal. At this point, when if we are more strict and we don't speak only the rodent, we take as a small group the subfamily Murinae. You can see in Europe, it's the number of this subfamily is very small, 6 species of Apodemius and 3 species of mouse and 1 microbes. And if we go to Spain, we have only 4 species plus the introduced ratus and mouse. It means, despite to have this very few number of species, we have worked for many years at this point. It means after past this time was necessary to move to new places to continue our studies, because our image, what we have in the Iberian Peninsula and the Atlantic and Mediterranean islands is quite clear. Also in Europe, it's many studies from the 50s from another teams. It means we need to find a new areas to explore. What will be these two new areas to explore? Then we will select areas with a diversity of Murinae and, of course, low number of students. And these areas, they consist. These areas with big number of Murinae have a low number of studies. For example, here I put the case of Africa. For example, if with total of 208 species of Murinae, and for example, if we take the case of Ethiopia, you can see that it's 18 genera. It means that if you compare with the previous, that for example, we say in the Iberian Peninsula, it's 4 species, it's completely different, and this is open a big chance to study. In addition, also in the Southeast Asia, it's the height diversity of these Murinae. For example, I put the example of Southeast Asia, 66 species. If we go to Wallacea, more than 50. and still the description of new species. It means, as you can imagine, compared with the previous studies that were done in Europe, these new studies in Africa and Southeast Asia mainly open many new possibilities to study. This is, for example, recopilatory of the literature data that, for example, if you pay attention, this is the number of new species described for science in Southeast Asia. We put this in groups of 5 years, and finally we found that we only have information in general, not only these new species. If we take all the literature that is involved elements of Murina and then in Southeast Asia, we will see that it's only 54 species that we have records of elements, representing only 20% of the species. It means, despite it's a continuous work, it's very small work, and it's a lot of to do it. In this case, of course, we are speaking about biodiversity of elements, of rodents, but of course other aspects like ecological aspects were also very understood. Screen, sorry. Okay, some examples that after this work I will center more in my normally in my work that is more taxonomical work. I will not include more other ecological papers that I was part of the papers, but I was much more people involved. I will speak more about these taxonomical papers that was my part of the work. In this case, for example, this is a new species of filaria that we described. It was from the genus Brenla. It was quite a lot of information of I don't see the different slides, says Johan. I don't see the different slides. No? Everybody can see? 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 Johan said that you are in the slide of Brenla. Can you see? Okay. Try the next one. Okay. This is another species that we describe, that we call this species Protospirura siamensis. So we make a description, morphological description of this species. And in this case, for example, is another species, Physalopteragnosi. In this case, it was the description in Vietnam in the year 661. And what we do is to redescribe the species because it was very few information. And also we increase the distribution of all these species, of these species, Physalopteragnosi, is distribution in Southeast Asia. It means that from this work, from the 61 that was in Vietnam, was nothing else information about this parasite, and with our work, we can see that was present in several of the countries. This is thanks to these extensive trappings that we did before in Southeast Asia, especially with Dr. Serge Moran. At this point, that as we tell, we have this intensive trappings, and we have all this series of information. Now I will speak things that they are in progress. In this case, for example, we take Heterachis spumosa, that is representative from the same group, for example, than Heterachis gallenarum. In this case, we have quite a nice collection from all Southeast Asia, and we pretend to compare. This species, the Heterachis spumosa, have a cosmopolitan distribution that is associated mainly with in Europe with invasive ratus. It means with ratus, this species become cosmopolitan and can be found every place. At the same time, is some endemic species like Heterachis daumensis that it was described by myself in Tanzania, and in this case can be found in a concrete species, Chrysetomis, in Africa. In this case, we were thinking of if Heterachis spumosa that we found in Europe, mainly in ratus, and we are supposing that this ratus, the origin area of invasion is Asia. What happens with the Pterakis in Sostethesia? We wanted to see in the origin of the species. Follow a little like the previous speaker about the invasive species. What we found that prevalence of Pterakis spumosa was not very high, less than 5%, but anyway, it was quite present in several of the examined localities. In the case of Ratus norvegicus, is underrepresented in the sampling in Sostethesia. We cannot get a lot of information. What we observed is that this parasite was found in several hosts, including Asparasidae. Means we didn't at present. It looks not a specificity. Was quite interesting because in Europe, as I told, it's mainly found in Ratus. Sometimes it in moose, Ratus and moose, that they are these invasive species. But normally in the native species is not very common to find. It means in the moment that we found all this, we thought that probably it was like different species of Pterakis in Sostethesia because the hosts were quite different compared with Europe. After to make all the genetical analysis, we didn't find this. In fact, we found that all the individuals that we found in Sostethesia, they form a symbol group, including different rodents at the same time, and the rodents with different habitats. It means that what happens here is looks not a specificity, and looks that this parasite is able to parasite several different rodents in several habitats, little different that we expected as we see in Europe. Another case that we studied was the case of Trichuris, concretely Trichuris muris, that it's quite interesting, despite that it's more than 250 years that we know this parasite, or also you can know for Trichuris serrata in cats or Vulpes in dogs. In the case of Trichuris muris, after 250 years, we think that everything was known. We know that this parasite is with a spread for Europe, for example, and another species, Trichuris ricoli, described for our group. It's found in Chrysetidae. Looks some sort of a specificity between Muridae and Chrysetidae. We continued these studies of Trichuris muris in the house mouse hybrid zone with the two subspecies of moose. And finally, about this, we studied this area between Germany and Prague, and finally, what we found is that it was not a specific clades. It means it was circulating these Trichuris. We think that this probably is due because despite the two house mouse, they are is isolated. It's other host like Ratus and Apodemus sometimes, and Apodemus that can allow to circulate this parasite. In this case, a difference of the previous parasite, Trichuris muris is also present in much more rodents. It means it looks the parasites are circulating. At this point, okay, the same case, we decided to see what is going to happen in Sostethesia to try to find more information about this Trichuris muris. We get some samples for ourselves, mainly from Thailand and also from Malaysia, and expecting to find this Trichuris muris. But after that, the more surprising was that in fact it was not Trichuris muris. We found a species from Borneo Island, this one in blue, and one from Thailand, compared with Trichuris muris, for example, and samples from Spain. It means that in fact we described two new species, one from Sostethesia, from Insular Sostethesia, and another from Borneo. These two species, the first one from Borneo is quite different for morphology, and the first one in Thailand is very similar to Trichuris muris. This can be the reason because during many years, no one saw this. It's also interesting that Trichuris muris, that it's a model used for lab, is not so much information in these aspects. And finally, what we can see. We can see clearly these two species. That looks at the geography makes that different between them. A little the same in Africa. Africa, we make some studies, and after, for example, in the same locality, we found we found the same parasite not sharing the same species. It means this is like interested in this case, be the same species, but looks like a specificity. And more studies also in Africa see us that in fact these 3 species is not one. One that I already described, Trichuris mastomici, another one that I described it from Gerbilis, Trichuris de planteri, and the other one that will have SP and moment that also is on the same course as Trichuris mastomici, but for the morphology it's very difficult to describe. It means we know that it's different species for genetics, but it really like officially make a new species because it's really difficult to make the morphological description. Also, we find other species like these ascaris that it's after the big surveying in Southeast Asia with Dr. Serge Moran with more than 4,000 rodents. Finally, we only found this ascari, Timberlimis bowersi, in 2 different localities. And also it was some all information of ascaris SP in Vietnam. It means it's like looks this very specificity just to compare with the other parasites that they looks that they can move more for different hosts. Okay, this is other species, pterodermatitis that this is still in process. More recently, we are working a little with Hymenolapis that this can be found in humans, Hymenolapis diminuta, Nana hibernia that have been recorded in humans, but is also shared in rodents. We try to make all the analysis of this big collection, expected to find Hymenolapis diminuta, Hymenolapis and Nana. But what we found, in fact, is the real Hymenolapis diminuta, but also we found 1, 2, 3 new species of Hymenolapis. In the case of Hymenolapis diminuta, morphological characteristics is difficult to see by morphology. It means with genetics, we see that in fact is several species that need to try to describe, but it's really difficult. Also for Rhodontelapis, Nana, also species in human. Also we found another clade. It means finally this group that we expect was very simple. In fact, it's at diversity, and probably a little like the previous study. These species are still in the original area, but many of them didn't travel to the new invasive areas of the or where is present, for example, Ratus, and this can be that explain the result that this information has been hid during long time, and since now we didn't get this information. The last case I will present is about the cat, about the teniformis. As you know, is the rodent intermediate host and the cat that we have the adults. In the first previous studies that we did it, we found in fact it was 3 different species. The Tigeretaniformis, that is the normal one that you can find in the books, and you find high diversity in Southeast Asia in the rodents. It means we can consider that this is the real origin of these species, and after move to other areas, because also we found in wild felids like Prionolorus in big distribution. After we found a second species in this study, that was Idatecher camillai, described like Idatecher camillai, and in this case is found in microtids, but mainly in north of Europe, like Finland, Norway, Sweden, Russia, and the final definitive cause is the either lynch. And the third species, it's only found in cats, in France, in Italy, and we don't know the intermediate host. Like example, the Metacestos, the larval form that is found in rodents. In Europe, it was only a single sequence from Spain. It means it was this situation that we found these 3 species distributed, the most common Tigeretaniformis, and the other 2 that are more restricted. In this case, we want to know If we have the clades in France and Italy and Camilla in the north of Europe, what happens in the middle? What we did it, we analyzed all the samples in the of several localities, the kist in mouse from Germany, Bulgaria, Czechia, France, Portugal, Bulgaria, from Mus domesticus, Musculus, and Mus macedonicus, and what we found, and also from Apodemus from Czech Republic in France, and we found that all these populations in Middle Europe is the one that we expect, that Ticheretaniform is associated with the domestic cat that have colonized all the world, but at the same time, the Tijera camillae that was only restricted to Northern Europe that we think was associated to lynx. Also, we found, for example, in France and in Czech Republic. It means, in fact, the two species are together in the same locality, and before everything was considered the Tijera teniformis. Yeah, for what is interested, don't mainly because this is the tenure of the cat, is supposed in many studies, and finally, in fact, we see that we expect that it's not true in fact is sharing to species. In conclusion, if we have this lack of information and in a species of veterinary interest or pet animals like cats, we have to imagine how much work have to be done for the elements on wildlife. This is like we do as we want, we try to advance, but we have work for many years. Okay, thank you very much for your attention.
It's very nice, impressive work in terms of taxonomy. Yes, we have one question for Professor Seven. And yeah.
Hey, Alexis, Professor
Yes.
Alexis. And
Yes.
yes, just let you know that we also recently do some parasites of wildcats in Taiwan. So if you need some samples, or you can tell us what kind of data you need, maybe we can send that to you.
Yes.
So you can do some comparisons. Yeah.
Yes, because we are like surprised that, for example, to find this species was supposed to be restricted to Northern Europe. We found like in the French Pyrenees together with Tenia teniformis. Also in Czech Republic, it means that two species are very with a spread in Europe, and we didn't expect this. It's like sometimes we get surprised, like as you tell, these samples from Taiwan can be another surprise. Who knows?
Yes, I suspect. Thank you.
Thank you very much. And I think because we are late, so now we're starting early, but finally now we are late, and we have quite a lot of several meetings still. And thank you very much, Alexis. And
Thanks
we
to
will go
you
to
for
the next
inviting me.
speaker.
Bye
Okay.
bye.
Thank you. So for the next speaker is Associate Professor Dr. Julia Court from ICM University de Montpellier, CNRS IRD, France. So his topic is Our Parasite, a temporal integrator of biodiversity.
So hello everyone. So my name is Julien Claude, and my presentation is about owls and rodents in Southeast Asia. So my presentation title is Owl Pellets, a temporal integrator of biodiversity, and I would like to associate Angeli Selim and Sylvie Gray for helping me for that presentation. So as you probably know, small mammal distribution are not well documented everywhere in Southeast Asia. To illustrate that purpose, here you have two maps. The map on the left side represents the occurrence of rodents available at the Global Biodiversity Information Facilities, the GBIF, that gather about 10,000 occurrences of rodents in the area investigated. So the distribution of this rodent is uneven. And if you look at the occurrence of rodents that you have here in GBIF, so you have to consider that you have the whole order Rodentia, meaning that you have a lot of squirrel. You have also occurrence regarding porcupines, and you have also occurrence regarding rats and mice. So if you want to have a picture of the recent records that were done by, I would say, naturalist people or people who are interested by this distribution, you can look at the record made by iNaturalist. So iNaturalist started to gather some attraction of the public in Thailand since about 1 year or 2 years. So it's rather recent here. And you can see that the place between iNaturalist and GBIF are matching well. This is because in GBIF, if you have a research level observation, things are reported from iNaturalist to GBIF. So GBIF is including the research level identification of iNaturalist. Not all the identification here are good. So for instance, several of these occurrences are related to Rattus rattus. And we know that Rattus rattus is probably not present in Southeast Asia, except probably in big cities where it can be considered as an alien species. So most of the time people write Rattus rattus, but this is Rattus tanizumi, the species that they have been identified or that they believe to have identified. So you have to keep in mind that more than 50% of mammals weight less than 100 grams. This part of biodiversity is not well monitored, and usually it retains less attention than large mammals. So when you have large mammals, people focus on the distribution of large mammals, and they don't focus very much on the small one. So this is the case in tropical countries where you have a very outstanding biodiversity. But in other places where large mammals are rare, so where the inventory for large species is better established, people interested in the distribution of small mammals. And this is the case, for instance, in France. So here you have a map showing to use the distribution of a single species of rodent, so glis that you have a picture here. And you can see that the occurrence for that species covered well the territory. So here the occurrence data match very well with the distribution of the species. While in Thailand, the occurrence data match well with where people have been documenting the biodiversity. So it's very difficult to know whether you have rodents or not, for instance, in northeastern Thailand, because there is no occurrence, but likely nobody was recording the rodents there. So in France, things are different because all the provinces try to record information regarding micro-mammal diversity. And this is done because we have national and local instruments for that. In particular, the government set up the national inventory for the natural patrimony since several years now, and the mammals are a target group. So in several places or several provinces, so I should not say province, but rather region. So in France, region are a groupment of several provinces. If you compare with Thailand, the inventory was done for micro mammals, so sometimes just rodents, but most of the time rodents and small other mammals. So, and in these inventories, you have a list of present mammals, but you have also an idea of their distribution. And the distribution is based on occurrence, which is not based on models. So most of the time, this inventory are possible because the government is stimulating this kind of inventory, and also because citizens feel more and more concerned by environmental and biodiversity questions. So you have an example of map produced at this province or regional level. Here, this is for Heliomys quercinus. So this is another small rodent that is well represented in France. And you have the map for the Burgundy region gathering for province. So you can see that on this map, the map is cut in piece of 10 square kilometer, and we know in each square whether the species is present or not. So when you have the green color, it means that the species is present. When you have the black dot, it means that it was present here but was not recorded during the present inventory. And if you have nothing, it means that there is no observation of the species. So, and this is done for all the regional in France, and this participates to the national inventory. So this inventory is eased because it is also including the analysis of regurgitation pellets produced by night birds of prey, such as owl. And this inventory is also possible because naturalist associative groups and sometimes groups working on owl pellet actively participate to the inventory and also to the identification of prey item in pellet. In this part of southwestern France, you have the distribution of the barn owl. So you can see that this owl is widespread and its occurrence is well documented because, as in many other places, so bird watchers and naturalists are more interested by birds than some other animals. And because we have a very good network of bird watchers, we have a fairly accurate idea of the distribution of the barn owl. If rather than to have local observation, we make a grid of 10 kilometer by 10 kilometer. So each round here shows you that we have at least one barn owl in one square of 10 kilometer by 10 kilometer. And you see that barn owls are nearly everywhere. So in the few places where they are not, so the inventory should be completed by personal observation or by investigation on the field. So these owls help to make the inventory. In some protocols, so people try to gather 600 preys for 4 squares. So meaning that they need on a square of defined on 20 kilometer to 20 kilometer of, there is a need of 600 preys to determine with some accuracy the micromammals communities. So this is what is said in the protocol. So if we look at Thailand, things are more or less the same. You can look at the distribution of rodents, so the occurrence of rodents on the left side. On the right side, you have the occurrence of owls and their relatives. So you can see that there is better efforts of sampling regarding the owl distributions and the rodent distribution. But you should know that every time that we have a owl, we should have some rodent or micromammal accumulation, because the owl will regurgitate pellet, and the pellet will contain an idea about the composition of the community not very far from the nesting place of the owl. There are several potential pellet accumulators in Thailand. So you have about 20 species, a little bit more than 20 species of owls in Thailand. Some are rare, like the eagle owl, but some are very common, like the 4 that you have here in front. So these 4, you can find them not very far from Bangkok, even in Bangkok, in some gardens. So for instance, this one here was photographed in Nonthaburi, in a place that is well known for bird watcher. And this is not difficult to find other owl in Bangkok at night. So these owls are particularly well represented in some environment. And for the barn owls, that is more or less the target of my talk today. So the barn owls is often present in caves. And in caves, it can generate big pellet accumulation. So the advantage of this pellet accumulator is this owl in particular hunt micromammals in various environments, and it can spray on small or even on large mammals. So remember, more than 50% of mammals weigh less than 100 grams. So the owl, like the barn owl, usually can take prey up to 80 grams, 100 grams, or even 150 grams. So owls can potentially record about 50% of the mammalian biodiversity. This is rather high. This is rather interesting, and this is why they are particularly interesting as a potential revelator for some cryptic biodiversity. So this is why, since 4 years, I am starting to investigate this pellet accumulation in Thailand. So here you have an example with a cave that I have been investigating in Mesai district, so not very far from the cave where the football team was lost. And in this cave, you can see that on the floor, we have some evidence of mammal bones. So these mammal bones, actually, they come from pellets that are disintegrating on the floor. And when you further investigate in the cave, you are able to find pellet, and mostly pellet of barn owls. So usually the rodent, the pellet of owls are found near the cave gates, near sky hole, or just close to the cliff, where the owls are doing their nest. They are never doing their nest very deep in the cave, and this is why it's rather easy and rather safe to collect this kind of accumulation. The two maps you have here are representing the rodents that were sampled during the Theropath and Biodiv health Southeast Asia during the I would say nearly more than last 10 years by Serge Morand team and other people. So I was part of few of this mission here. And you can see that this trapping session, we are covering also part of the territory, but the distribution is also uneven. And since the Future Health Southeast Asia project, so I have been starting to document the occurrence of rodents based on wool pellet. And you can see that you have this wool pellet site in yellow on the map. So meaning that sometimes we find wool pellet very close to sites we were investigating, like for instance in Loi, Innan or Chiang Rai or Mae Son. And sometimes we are able to find pellet in other places that were surveyed by trapping session. So meaning that we should be able to compare biodiversity by the mean of traps and by the mean of owls in some place. And in other place, we can document the composition of rodent communities just by the mean of the owl. So this is interesting because rodent bones and teeth can be accumulated also on several years and can fossilize. So this gives us access to retrospective data. And this kind of retrospective data can sometimes be rather old. So for instance, in Mae Son, sometimes the accumulation can be found together with archaeological remains, such as these boats that were used in the cave as grave in the past, or sometimes directly in the sediment, where you can find accumulations here of shells together with rodent bones. This is the case here in Chiang Rai. And sometimes the accumulation of rodents can be impressive and can be on several centimeters, decimeter, or even meter. So here you have an example of bone accumulation in Chiang Mai. The floor is completely covered by rodent remains. So all these small items here, these are incisor of rodents, molar of rodents, and small bone of rodents. So this is not the first time that people are studying wool's pellet in Thailand. So for instance, you get studies that was done. So this is said to be a first investigation on diet of wool based on pellet in northern Thailand. And as you can see, we still need to do more research in this direction. So in this 6 years old study now, people were determining the rodents in wool pellet by using molecular techniques, but mostly they have found Rattus rattus or Mus musculus. And we know that these species are probably not present in the area. And by comparison, they were collecting very, very few local species. So there is a need to produce local reference. So for DNA, of course, so these references are on the way to be produced for several mammals, but also for anatomy. And this is essential because if we want to get the data on 600 rodents at every 20 by 20 square kilometer and employ naturalists and citizens, one might start to seriously think about some alternative to DNA barcoding. So this is why we are starting to do pellet preparation, which is rather simple. take your pellet, you soak it in water, you separate the bones, then you clean the bones. You make the inventory, pellet by pellet, you take pictures of skull features, and sometimes you do morphometrics to help you to identify the bones. So the pellet work chain here is made more complicated because we do not have still good references for identifying the bone items. So we have to define skull character. We have to define skull and teeth characters from Voucher specimens. So for rodents, we have a good collection now with the former trapping project, and because many of these rodents were obtaining a barcode. But for other mammals, things we'll have to follow their way. I think, for instance, for the Insectivora or for the Chiroptera, we'll have to keep in touch with molecular specialists to help us to make the first databases. Then we identify items in pellet, and the advantage of pellet is that we can associate skull with other bones. So meaning that after we will be able to identify specimen based on skull features, maybe if we are lucky, we will also be able to define features on other part of the skeleton, for instance, limb bones, to appreciate the species composition. And at the end, this will allow us to have a tool also to determine fossil or subfossil material where the pellet are completely disintegrated and where we just have a part of the jaw or just a few teeth to identify the specimens. So this is important if we want to establish long terms or middle terms diversity spatiotemporal dynamics. So we started to look at what we had in the pellet, and in the pellet that we were collecting, we do not have any mammals, but we have also reptiles. So they are not a dominant part of the pellet, but they are present, such as lizards, snakes, sometimes amphibians. So in the pellet, we can sometimes find birds that are not, we don't find them often, but they occur. Some other things are more difficult for me or for my colleagues to identify. So this small piece here may be likely a piece of fish. So it's possible that in caves, sometimes fishing hools can also accumulate regurgitation pellets. And fishing hools can accumulate fish biodiversity, even if it's difficult to find this kind of hools. Of course, we can find mammals, some mammals belonging to different orders. So rodents, of course, but Insectivora are items that we often find in the pellet, small or big. There is also a large array of Chiroptera prey, so the very small one from the very small one to the large one. So including also fruit bat. So you don't have here pictures of fruit bat on the slide, but they are also present. And for rodents, we have representative from the very small one, like the mouse, to the large one, for instance, like this jaw of probable Rattus norvegicus that was trapped by a barn owl. So, and for some species, we have a very good series. We can have an ontogenetic series, or we can have intraspecific variation series that will help us to define the variation of morphological traits for a given population of rodents. So there is a need to produce more accurate taxonomic keys and to produce these keys for potential naturalist association and for citizens. So this is the hard part of the work. So this is to produce keys for taxa for which biodiversity has been largely challenged by molecular analysis. This will take times, likely several generations. So I will not do that alone. But it's worth trying to start it as the accumulation of pellets will continue to accumulate in times. And as exploring the biodiversity by that means is less intrusive than to any other kind of research where you trap the rodents or where you manipulate them. So once we will have identified the whole communities, and there's an important part of work to do here, with the pellet that I gathered, there is 15,000 to 20,000 rodents to identify so far, rodents on Insectivora on bats. So we can probably look at the sampling bias between hools and trapping methods because there has been intensive tapping session during the last 10 years. We can look at community composition on environmental variation. So you should know that microvertebrates are good bioindicators. These are sometimes used for paleoenvironmental reconstruction. We can also look at community dynamics and dynamics of evolutionary dynamics in these communities, if we look at morphological traits, for instance. We can also look directly at the evolution of the ecology of these rodents in these different communities by looking at other proxies, like their position in the trophic chain or their diet by using stable isotope, looking their diet by using dental microwear patterns that can help us to know whether they are more eating fruits or seeds or whether they are eating more herbs or things like this. And of course, morphometrics can also help us to relate the morphology with environmental variation here. So to conclude, all pellets are present in Southeast Asia. In some places they are abundant. So pellets that we were examined so far shows that the birds are not really selective regarding prey item like in other places. So meaning that we have good biodiversity indicators here. So there is a hard work to furnish to establish new taxonomic case because we don't have any rodents here, but we have plenty of other mammals. So this is or sometimes also reptiles. So there's a lot of work to produce here. And as this pellet can be informative, we should now investigate other sites and caves. And for that, we need probably the help of citizen and association, I think mostly about bird watcher. I would add that pellet dissection as well as participatory taxonomy is things that you can easily teach in school or high school. This is also a way to raise the awareness of future generation on biodiversity management and biodiversity management. So to finish, I will thank the different sponsors of my research. So the IRD and the National Research Agency, my partner in Bangkok, Chulalongkorn University and Sipakorn, as well as international network regarding paleobiodiversity in Southeast Asia. So thank you all for having listened to me, and bye-bye. Serge, I cannot hear you. I think your microphone.
Okay, so I restart again. Sorry, I was mute, not by myself, but by the microphone of the room. Yes, I would like to thank you very much, Julien, for this talk, and very interesting. And it echoes me what we listened this morning with the talk of Dr. Rodolphe using mosquitoes as a way to sampling biodiversity and sampling pathogen. But with your approach, is much more, is broader because you can have also the remaining of these animals with the skulls, with the bones. So you can investigate, of course, the DNA, but you have also the morphology, but also with the remaining in the caves, you can have also the past. I think is very fascinating and very innovative, and is really complete a lot of the biodiversity inside. And I would say that if you investigate the DNA in the bones, you can also investigating some pathogens that still remain, especially the bacteria that can be associated with the blood, that we can also even potentially investigate.
So far, and unfortunately for you, Serge, we are eating the bone before to send them because we want to destroy all the pathogen, all the bacteria and everything, because as we are manipulating this pellet from very close, we want to avoid any problem with fungi and bacteria.
No,
So
but
best-
this is a way, of course, but you have a way to every year to manipulate. So it means that it's like when you are digging the bacteria in the soil, you can isolate it on the field and after send back to some lab that are really specialized for this. I did not ask you for you to do this or to put you, just isolate the specimen like we did for several sampling of animals.
Okay, no
Anyway.
problem. I can do that.
Yes, would be great. Is there some question in the chat?
Yes, they have one question from the message. They ask, can we know the rodent population dynamics from our palette if we collect maybe 5 or 10 data in the particularly area to speculate population dynamic of prey species?
So I hope that we can, but owls as humans produce bias. So some year they will sometimes focus more on some part of the landscape than some other. So this is why I'm interested to compare also the composition of what we got from owls and the things that we got from traps. I think in the middle term, and by doing some averaging, we will have trends. And one of the good advantage with barn owls is that usually if you don't disturb them, they will come back on the nesting site nearly all the time. So meaning that you can monitor a place for 20 years and you have a natural observatory here of, and this is rather cheap because you just have to collect the pellet and just be careful not to annoy the owl to come back again.
Thank you very much.
Okay, no question. Okay, so we will go ahead from the next speaker. The next speaker is Dr. Pipat Soysuk from Princess Mahachakri Sirinthorn Natural History Museum, Prince of Songkhla University, Thailand. He going to talk about Bat of Thailand, diversity and conservation.
Hi there. Hello everyone. Thank you for joining me with this talk, and thanks to the organizer, particularly Professor Serge Moran, for inviting me to the seminar. My talk going to be very simple about bats of Thailand, trying to give you a bit more insight about the pattern of diversity, the work that we've been trying to understand more. I mean, my work in terms of taxonomy and our little effort in bat conservation that we're working with researchers either in the country and also with the researchers around the world. And as you know, bat is very highly diverse. It's the second most diverse mammal. Let's start with this picture. This bat in the slide. I'm not sure if you can recognize them all. Even just one or two, that's good enough already. So this is the picture of the bat. They've been trying to take a good one to, you know, make the public outreach, trying to making people understand more about bat correctly, rather than understand that bat as the origin of the COVID-19, blah blah blah. But instead, bat is our friend, and they deserve to be protected. So let's try this one. There are some of them I'm sure that you can recognize, and some of the Thai bat and some of those from Africa, from America, and some around here. Some of them are very popular, like Yoda bat in the bottom right corner. So there are lots of bats out there. So one of the things is we're trying to understand why there are so many bats in the world, and what is the pattern of this diversity and how can we conserve them properly. So my message today is that whatever you do and whoever you are, you're probably working on disease ecology or just ecology, biology, ecolocation, any disciplines about that. I would say at the end of the day, you're gonna need to work more or less about conservation. So we're gonna need to work together on that. Whatever you are expert on. So let me introduce some of you that not very familiar with that. So what we're working on is this very diverse mammals, and it's found from around the world except the Antarctica really, and there are more than 1,440 species currently described, and it's increasing very rapidly, particularly from the tropical zone like South America, tropical Africa, and Southeast Asia here. So if you are looking for the literature, the book that contain the most updates, the species of bats around the world, is this book, The Mammals of the World, volume 9, dedicated to order Chiroptera. And the web page is quite very fantastic, I think, is batnames.org. Take a look at that website, and you can see the real-time updates about taxonomies of bats around the world and the comments. And here is the basic classification and phylogeny of bat. So we got 2 suborders, Enterochiroptera and Yankochiroptera, and the bat originated from about 52.5 million years ago, with the closest relative is those carnivores. So not only species that's very, very diverse, the habitat and also the behaviors of bats are also very diverse as well. Bat can be found in colonies like this flying fox, in the trees, or the big colony as well, but inside the cave, like this cave nectar bat, the one that's really, really important in terms of pollinating a lot of flowers like durian, petai. They probably roost like this under the leaf as a harem of one single male, and they're very important in the ecosystem. They are keeping the balance and dynamic of several ecosystems, such as the forest, and also give us the benefit in the orchard as well. So here's the picture of Cape nectar bat pollinating the durian flower, which it turned out that we got billions of dollars per year of durian, and that is just only because of we have this bat around. And they are also very good in planting the forest, so we don't need to do a man-made plantations or reforestation or anything. Just we only need to keep the bats around us, so then let them do the job, so they disperse the seeds for us, and then we get the forest back. The bat can also help us suppress the insect, for example mosquito. So this size of bat is around 2 or 3 grams, something like that. It can eat up to 1,000 mosquito in an hour. And also they help controlling grasshopper. This is the pest in rice field. As you know, Thailand has a lots of rice field, and imagine this: 2.6 million individuals of bat at Khao Song Pran cave in Ratchaburi province. How many grasshopper and insect that they help eating and then save the rice field. So millions of tons of rice have been saved by this wrinkle-lipped free-tailed bat. So again, there's a long constant sound, like that. The short one is from the Hippostyros right here, but the long one is from here, the Rhinolophus or horseshoe bat. So let's hear from this Myotis, the one that is known as FM bat, frequency modulate bat. So it's gonna be pretty short click right there. It's a very quick sweep sound. So, and we got the frequency and the characters of the sound of each species. Then we can recognize them even without capturing them. One of the jobs that we're trying to do at the moment is to get the database of the echolocations of bats around the country. And the next amazing thing is that bats have a very long lifespan. So the longest record is that 43 years is from Myotis brandtii. So this is one of many things that's really amazing about that. Besides that, there are only one flying mammal. So when you look at the size and the body mass, comparing to the lifespan, that is the very long lifespan comparing to the other mammals. Come back to the Southeast Asia and Thailand. So we are at the very, very highly diverse hotspots of bat in the world. So around only ASEAN, we got almost 400 species of bat known at the moment, and that more than 50 of them are only discovered or named or elevated to species rank during the last 15-20 years. One of the reasons is that we got network, we got more researchers coming up every year, and we got the very, very complex biogeographic patterns in Southeast Asia, besides having a lot of suitable habitat. But at the end of this talk, I'm gonna point out what's gonna happen if we are not trying to conserve the habitat and the bat. So what about Thailand? What do we know? At the moment we got, well, at least in my list, we got 146 species from 11 families and 46 genera. The most diverse family is Vespertilionidae here. And when we divide them into the habitat, we can divide them roughly as cave bat and forest bat. It's about half of them, 49%, are forest bat, and about 44% are cave bat. And then the rest, about 7%, are those who can adapt to the orchards or the urban area. We got only one official endemic species at the moment, which is Pendlebury leaf-nosed bat. It's only found in Peninsula Thailand, with about 8,000 individuals known at the moment, and is listed as vulnerable species. We also have the other species as well, but I guess they will be proved to be found in the other countries as well, like Chrysonectris, the kitty hawk-nosed bat, that is already found in Myanmar for almost 20 years, I guess. And the Eudiscoderma, Tongaridis node bat, which is the new genus that we described 6 years ago, and I guess we can be found. We can find them in Malaysia as well. And as I mentioned, we got a lot of unknown species around, not only in Thailand but in Southeast Asia. Once we work together, we can solve a lot of taxonomic problems like this one that we found in Borneo and also Thailand and Vietnam. Then we pool together all of the information of this group of horseshoe bat from India, from Taiwan, from China. Then we come up with the description of the new species like this one. So take a look at the biogeographic pattern of the bat in Thailand. So basically we can divide them into the Indo-Chinese group and Sunday group. So roughly bats in Thailand distribute by following this pattern, divided at the Isthmus of Kra. So the upper part of the countries are one group and not overlap. The species that not overlap with the ones in the south. For example, this one, the tube-nosed bat, genus Murina. So there is no overlap at all. The Indo-Chinese species and Sundaic species. There's only one exception, is Murina hattani. There's one single record from Malaysia that I believe it is a different species. We are working on it now. But not all of them following this pattern. The Indo-Chinese Sundaic group, we found that the bat in the group of Pipistrelle, for example, Pipistrellini, like this one, Eudiscopus, or this foot bat, it is known as Indo-Chinese species. It's very rare one, but we find them in the south. So I guess the Pipistrelle, the group of bat in the genus Pipistrellus, Hypsugo, and this guy, they are more diverse and then more widespread than we thought. We started to find them in the south now, and we are looking at the population genetic in terms of even they are the same populations or they diverse from the Indo-Chinese population down to the south, or they have the refuge down here for a long time ago already. And this is another example. We found the Indo-Chinese thick-thumb bat from southern Thailand as well. So it's probably proof that it is a different subspecies or something like that. But our pilot study saying that they are pretty much the same with the Indo-Chinese population. we're looking at the skull, the teeth, comparing all of them with all of the species that we know from Thai Malay Peninsula. Looking at the baculum and all of the evidence like genetic. So we might prove that some of them are the different subspecies, you know, the one from the south and the one from the upper part of the country. They're probably different subspecies, but in some cases, but they probably are pretty much the same. But this is one of the exceptions in terms of biogeographic pattern. The pipistrelle, like this genera, they can be found overlapping between in the Chinese and Syndic subregion. But there are a lot more taxonomic problems out there waiting for us to solve, like flying fox. Like this one, we trying to solve that whether we have Pteropus intermedius, the one that's very, very rare and known only from Myanmar. Either it is distinct or really exists, or it's just the synonym of the other species, like Pteropus medius or giganteus, or something like that. So the students from Myanmar are working on them and trying to get the information and the data from the museums around the world. And also this small Hipposideros, this is really widespread in Southeast Asia and very, very complex in terms of taxonomy and really hard to identify. So we probably get like quite a few more new species and new record that you know, it probably haven't been found for a long time. And then after we review the whole group, pulling out the information from the whole region, and we can come to the answer about the pattern of diversity of this small Hipposideros. So here's the ongoing taxonomic study that we are working on. It's not only our interest about taxonomy, we also work a lot with local community. Like here, we work with Stone UNESCO Global Geopark in Stone Province, and one of the interests is the bat, because we got the assignment to examine the diversity in the cave. So as you know, bat is one of them. It's one of the most important organism in the cave. So we find the huge diversity of animal, vertebrate and invertebrate, from the cave down there, and then it breaks the record of the largest number of species known in the cave. And this information going to be used as the basic data to manage the geopark in the future. Return all of the information back to the local community to use as the you know, the data to provide to the tourists, making the ecotourism in the areas more effective, you know. And then that is something that we talk about, the sustainable tourism. So we come up with some of the example that the local guide can use as the key animal that they can use, you know, to guide people, the tourists, to know more about wildlife in the cave. Not only vertebrate, invertebrate as well. Some of them hardly to find, but with the information of the researcher provide, then it is more to be accessed by the tourist and the guide, and then they can understand more about the animals in this area. We also come up with the information about the mitogenome of the endemic bat that I mentioned earlier, the Penderbury leaf-nosed bat, Hippoceros penderburyi. We got the tissue sample and sequence the mitogenome from Geopark, and then we're going to publish the mitogenome of this bat for the first time. This is one of the research that we're working with local people. But at the same time, we should not forget that the hunting of bat still occur out there around the country. People still eat bat, particularly with alcohol, you know. So just one catch, many of them are gone. So they don't care about the benefits of bat. They just want to eat them. It is not the main food for their daily life, but it's only just, you know, for the snack with booze, just like that. So they hunt them and eat them. So we're gonna need to come up with a campaign to help conserving bat, not just only doing research on them. The CB crew, for example, is one network around here. It's working together from all of the ASEAN countries in different research focus, and taxonomy is one of them. But the major goal is to help conserving the bat in Southeast Asia. The bigger network that includes CB crew at the moment, that you probably heard already, that we have GBATnet, the global bat network, putting up together all of the bat networks around the world to work together, then encounter the misunderstandings about that, and also put together the major goal, the common goal toward the bat conservation. So check it out, GBATnet website. So as I mentioned, we were not only doing the basic research of our interest only. Like myself, I work on taxonomy, but at the day, as I mentioned, we're gonna need to work on the other thing as well, not just the research that we love to. So we need to do the training, we study the other aspect as well, particularly understanding the development that probably affect the bat populations, like the wind farms. And my idea is to work with every level, from the top of the country, like the policy makers, and also the research. But at the same time, we need to work with local people as well, because they can give you a more effective way and sustainable way to conserve the bat. So my museum work with local community around southern Thailand to provide the expertise on biodiversity, and I have my part on that in terms of bats and small mammals. And one last thing that I would like to say again is that we're gonna need to do the other conservation activity as well, not just the research. So we need to start doing more public education and public outreach, talking more to the student and the public to make them understand more about that, not only just let them understand that is the origins of COVID and there are scary, those kind of things, but make them understand how important and what benefit we got from having bats around us. All right, we probably need to put something like the activity, like the Halloween night, to talk about the facts about that, like I did here in the Halloween, so many other stuff that we use, the book, the nice pictures of the bat, the mascot, the T-shirt, and everything we're trying to put on, and then trying to use this stuff to get closer to the children and the student, and then let them be the medium to learn more about bats, and then they understand more about it, and then they will be part of the network to help us conserve the bat. That's my goal and my main message I would like to make today. So thank you so much, and we probably have a few minutes for the question, I guess. Cheers. Thank you very much.
Serge, you have been mute again.
Sorry, I am mute again. Thank you. And as I said, that thanks a lot for Dr. Pipat, and this is the best advocacy for bat conservation I ever have heard. Very, 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 very nice. And as you say that you work at the different levels, from the local, from the children up to the policy makers, amazing. But doing also a very good high level of your of research. You are one of the we are among the top researchers in bat ecology on bats, which is mean that you can do all good science, but also good translation of science on working to bats. I would like really to congratulate you for that. If there is any question, but I'm not sure that Dr. Pipat is with us. Yeah, no, he's not there. Okay. So we can keep, but anyway, you have everybody, so you can keep some questions, and after send to we can send to Dr. Pipat without any problem. So we will be very happy for this, and we can go to the next speaker. Thank you.
For the next speaker is Dr. Alexis Libas from Wildlife Conservation Society, Vietnam. The topic is bats, rats, and coronavirus in Southeast Asia.
Hello, everyone. I am Alexis Libas. I'm a wildlife health technical advisor and research scientist at the Wildlife Conservation Society in Hanoi in Vietnam. And first of all, I would like to thank the organizers for giving me this opportunity to present my research. So my talk today will focus on bats, rats, and their coronaviruses in Southeast Asia. So I will talk about how these coronaviruses evolved and diversified in this host. So just a quick introduction to start. Coronaviruses are RNA viruses causing respiratory and enteric diseases in humans and animals. They are characterized by the largest genomes among all mammalian viruses, They are also characterized by high genome plasticity, and they are prone to very frequent recombination. Coronaviruses are currently divided into 4 main genera: the alpha, beta, gamma, and delta coronaviruses. Alpha and beta coronaviruses mostly infect mammals, while gamma and delta coronaviruses primarily infect birds. So my talk today will mostly focus on the alpha and beta coronaviruses infecting bats and rodents. All known coronaviruses currently known to infect humans are zoonotic or of animal origin, and many of them originated in bat hosts. So bats harbor the largest diversity of coronaviruses among mammals, and 2 coronavirus genera, the alpha and beta coronaviruses, have been widely detected in bats from most regions of the world. Bats have been identified as the evolutionary sources of SARS coronavirus 1, SARS coronavirus 2, and also MERS coronavirus, the Middle East Respiratory Syndrome coronavirus. And bats are also the source of progenitors for the human coronavirus NL63 and 229E. On the other side, rodents were identified as the evolutionary sources of the human coronavirus OC43 and HKU1. And as you can see on this figure, a large diversity of intermediate hosts have been identified and were involved in the emergence of these bats and rodent viruses in humans population. Pangolins here have been suggested at some point as potential intermediate hosts in the emergence of SARS coronavirus 2, but it has then been demonstrated that was not the case, and they are not directly involved in the emergence of this virus in human populations. This map from a preprint by Baker et al. Shows that shows the species richness of known bat hosts of coronaviruses. And you can see that bat hosts of these beta coronaviruses are found worldwide. They are in Latin and Central America, but also in Africa and in Europe. But as you can see on this map, it's in Asia that we observe the highest species richness of bat hosts of beta coronaviruses. And this region, including China and Southeast Asia, is a real hotspot of bat host of beta coronaviruses. So China and Southeast Asia are characterized by a large diversity of both bats and coronaviruses. 3 coronaviruses emerged in China in the last 2 decades. SARS-CoV-1 emerged in southern China in 2002. And then, as you all know, SARS-CoV-2, causing the COVID-19 disease, emerged in China in 2019. But in 2016, another virus, the swine acute diarrhea syndrome, or SADS coronavirus, emerged in domestic pig populations in southern China and caused the death of more than 25,000 pigs in the region. So all these 3 coronaviruses that emerged in humans and in livestock have originated within horseshoe bats or the Rhinolophus bats. And we know that other coronaviruses with potential for zoonotic emergence have been identified and discovered in China and in the neighboring countries. So these findings highlight the importance of coronaviruses of bat origin to global health, and therefore understanding their origin and cross-species transmission is a high priority for pandemic preparedness. So my talk today will be divided into 2 main sections. The first one will focus on the origin and cross-species transmission of bat coronaviruses in China. I won't talk about the origin of SARS-CoV-2, but I will discuss the macro evolution of these coronaviruses in their bat host in China. And then the second part of this talk will focus on rodent coronaviruses along the wildlife supply chain in Vietnam. So in this study of bat coronaviruses in China, we analyzed an extensive data set of bat coronavirus sequences from across China, including more than 1,200 sequences of the RNA-dependent RNA polymerase gene, or the RDRP gene. And in the study, we used a phylogeographic Bayesian statistical framework to reconstruct virus transmission history between different bat host species, and we also reconstructed the virus spatial spread over evolutionary time in China. And then we also used estimates of phylogenetic diversity and phylogenetic clustering. So our objectives in this study were to compare the macroevolutionary patterns of alpha and beta coronaviruses and identify the host and the geographical region that act as centers of evolutionary diversification for bat coronaviruses in China. And all these analyses aim to improve our understanding of how coronaviruses evolve, diversify, circulate, and transmit between bat families to help identify bat host and also geographical region where the risk of coronavirus spillover is the highest. So we used a Bayesian discrete phylogeographic approach implemented in the software BEAST to reconstruct the ancestral host of each node in the phylogenetic tree. So this is here the phylogenetic tree for alpha coronaviruses. And you can see that Chinese bat alpha coronaviruses were identified in 5 bat families: the Hyposideridae, Miniopteridae, Theropodidae, Rhinolophidae, and Vespertilionidae. And this phylogenetic tree suggests an evolutionary origin of alpha coronaviruses within Rhinolophid and Vespertilionid bats. And the first alpha coronavirus lineage to diverge historically, here, this one corresponds to the subgenus Rhinacovirus that is originating within Rhinolophid bats. And it includes sequences related to the HKU2 and SARS coronaviruses. Then you have several lineages that emerge from Vespertilionid bats here. And the first one to diverge is this one that includes sequences mostly associated with the Rhinolophid and Hyposideridae bats, and they are related to HKU10. And then you have here all the remaining lineages includes coronaviruses mainly from Vespertilionid and Miniopterid bats. So all these 7 alpha coronaviruses lineages are mostly associated with a single bat family, but each of these lineages also included several sequences that were identified from other bat families. So that suggests that frequent cross-species transmission events have occurred among these bat families. So now here is the phylogenetic tree for the beta coronaviruses identified in bats in China. And these beta coronaviruses were identified in only 4 bat families: the Hyposideridae, Theropodidae, Rhinolophidae, and Vespertilionidae. So they likely originated from Vespertilionid and Rhinolophid bats. And this phylogenetic tree is clearly structured into 4 main lineages. The first one to diverge is the lineage C, or Myrbecovirus. It includes the MERS and HKU4 coronaviruses. And then you have this lineage D, Nobecovirus, that originated from theropodid bats and includes the HKU9 coronavirus. And then we have here the lineage E and the famous lineage B, the Sarbecovirus, that were isolated in Hyposideridae and Rhinolophid bats. And this lineage B, Sarbecovirus, includes the SARS-CoV-1, SARS-CoV-2, and the HKU3 coronavirus. We also used Mantle test to assess the correlation between the genetic differentiation among bat coronaviruses and the phylogenetic distance among their hosts. And we detected a positive and highly significant, as you can see here, highly significant correlation between genetic differentiation among beta coronaviruses and the phylogenetic distance among their hosts. But we detected no correlation at all between the genetic differentiation among bat alpha coronaviruses and the phylogenetic distance among their hosts. So these findings revealed a significant effect of host phylogeny on beta coronavirus evolution, while this had very minimal effect on alpha coronavirus evolution. As you can see when comparing these two figures here, contrary to the alpha coronavirus phylogeny, the basal phylogenetic structure of this phylogeny tree for beta coronavirus mirrored the most recent phylogeny of their bat host, with a clear distinction between the Yango-Chiroptera, which includes the Vespertiliodid and the Miniopterid bat families, and then the Yin-Pterochiroptera, which include the megabat family Pteropodidae, and then the microbat families Rhinolophidae and Hyposideridae. So these findings suggest a profound co-macroevolutionary process between beta coronaviruses and their bat host, while the phylogenetic structure of alpha coronaviruses with these numerous and closely related lineages identified in Vespertilionid and Miniopterid bats contrast with the beta coronavirus macroevolutionary pattern and suggest that alpha coronaviruses undergone an adaptive radiation in the Vespertilionid and Miniopterid bat families. We used a Bayesian stochastic search variable selection procedure to identify significant viral host switchings between bat families that occurred along the branches of the phylogenetic tree. And we calculated the Bayesian factors to estimate the significance of these switches. We identified 9 highly supported interfamily host switches for alpha coronaviruses, 6 of them involving the Rhinolophid family. And we identified only 3 highly supported host switches for beta coronaviruses in China, 3 of them involving the Hyposideriid family. Then to quantify the magnitude of these host switches, we estimated the number of host switching events, and we estimated the rate of interfamily switching events per unit of time for each coronavirus genus. And you can see that the rate of interfamily host switching events was 5 times higher for alpha coronavirus with 0.01 here, 5 times higher for alpha coronaviruses than beta coronaviruses, where the rate is only 0.002. And when you look at these figures here, these 2 figures, you can see the number of switching events from and to each bat family. For alpha coronavirus here, host switching events from the Rhinolophid and the Miniopterid bats were greater than from all other bat families, while Rhinolophid bats here were the highest donor family for beta coronaviruses. Then the Rhinolophidae and Vespertilionidae for alpha coronaviruses and the Hyposideriid family for beta coronaviruses received the highest number of switching events. So in summary, our findings revealed a significant effect of host phylogeny on beta coronavirus evolution, while it had a very minimal effect on the evolution of alpha coronaviruses. Our ancestral host reconstruction and analysis of host switching events demonstrate that a significant amount of cross-species transmission has occurred among bat hosts over evolutionary times. And larger numbers of highly significant host transition and higher rate of host switching events were inferred for alpha than beta coronaviruses. So these findings suggest that alpha coronaviruses are able to switch hosts more frequently and between more distantly related taxa than beta coronaviruses, and that phylogenetic distance among hosts represent a higher constraint on host switches for beta than alpha coronaviruses. We also found that bats in the family Rhinolophidae, the horseshoe bats, played a key role in the evolution and the cross-species transmission history of alpha and beta coronaviruses, this family being at the origin of most inter-family host switching events. There is a large diversity of rhinolophid species in China and Southeast Asia. So this map here shows the modeled distribution of 49 rhinolophid species. And you can see that rhinolophid hotspot occur in forests throughout much of mainland Southeast Asia, but the largest hotspots are observed from southern Laos and southern Vietnam to southern China. So this is the region where the diversity of rhinolophid species is the highest, with a maximum of 23 species known to occur concurrently. And these horseshoe bats are known to share roost with genera from all other bat families included in this study. So this co-roosting may favor cross-species transmission of coronaviruses from and to rhinolophid bats. We then used a similar Bayesian phylogeographic model with biogeographic regions as character state to reconstruct the spatio-temporal dynamics of coronavirus dispersal in China. 6 biogeographic regions were defined within China based on the similarity of their mammalian diversity. So these 6 regions are the northern China, central northern China, central China, southern China, Hainan Island, and southwestern China. And we identified 11 highly significant dispersal routes within China for alpha coronaviruses, and we identified 7 highly significant dispersal routes for beta coronaviruses in China. And on these 2 figures, darker arrow colors indicate older dispersal events. So you can see that the oldest dispersal events for alpha coronaviruses originated from the southwestern and southern region. And then dispersal event occurred in many directions in China. The oldest dispersal events for beta coronaviruses occurred among the southern and southwestern regions. Then several dispersal movements likely originated from the southern and the central region, and more recent southward dispersal from the northern region was also observed. The estimated rate of migration events per unit of time along these dispersal routes was more than two times higher for alpha coronaviruses than beta coronaviruses. It's 0.02 or 3 for alpha coronaviruses, and it's only 0.01 for beta coronaviruses. And this figure here shows that southern China, in blue on this these figures, southern China was involved in the greatest total number of migration events, both outbound and inbound migration events for both the alpha and beta coronaviruses. We then also assessed patterns of coronavirus phylogenetic turnover or phylogenetic differentiation among Chinese biogeographic regions by measuring the inter-region value of mean phylogenetic distance. And so based on these values of mean phylogenetic distance, we then used phylogenetic ordination to assess the phylogenetic relatedness among bat coronaviruses from different biogeographic regions in China. And our phyloordination among biogeographic regions reflected the high phylogenetic turnover and the deep evolutionary distinctiveness of both alpha coronaviruses and beta coronaviruses from southwestern China and the Hainan region for alpha coronaviruses, while coronaviruses from northern and central China were closely related for both alpha and beta coronaviruses. So in summary, these geographic analyses show that southwestern and southern China act as centers of diversification for both alpha and beta coronaviruses. And these 2 regions are hotspots of coronavirus phylogenetic diversity, and they harbor evolutionary old and phylogenetically diverse lineages of alpha and beta coronaviruses. These regions are characterized by a subtropical to tropical climate, but also dense growing and very rapidly urbanizing populations of people, and also by a high degree of poultry and livestock production. So all these factors may promote cross-species transmission and disease emergence. We did not find any significant correlation between coronavirus phylogenetic diversity and bat species diversity for each biogeographic region. So it seems that bat host diversity is not the main driver of coronavirus diversity in China, and that other ecological and biogeographic factors may influence this diversity in China. So now the second part of my talk will focus on rodent coronaviruses detected along the wildlife supply chain in Vietnam. So the live rat trade is highly developed in Vietnam. In 2005, the Vietnamese field rat trade was estimated to process around 3,600 tons of live rats annually for human consumption. And this human-wildlife interface involves the capture of wild field rats and then the subsequent trade and consumption along a supply chain involving the entire Mekong Delta region, but mostly Cambodia and Vietnam. So for this study, the Wildlife Conservation Society sampled live field rats, mostly from the Rattus and Bandicota genera, at 3 interfaces along the live field rat trade. That included rats sold by rat traders, but also by vendors in large markets, and then rats butchered and sold in restaurants for human consumption. Then we also sampled rodents raised on wildlife farms, so it was mostly porcupines and bamboo rats, to assess the risk from different wildlife supply chain for human consumption. So in total, we collected more than 1,500 samples, and all of them were tested for coronaviruses. And as you can see here, a third, 33% of these rodent samples were positive for coronaviruses. And we detected 2 rodent coronaviruses, the murine coronavirus and Longquan coronavirus, at the interface that were included along the rat trade. But surprisingly, we also detected a bat coronavirus, the bat coronavirus 512, and a bird virus, the infectious bronchitis virus, in the rodents, the 3 rodent species that were sampled in wildlife farms. So this indicates coronavirus cross-species sharing at this interface. Another interesting result that you can see here is that we observed an increasing proportion of coronavirus positive along the distribution chain. As you can see here, only 21% of rats sampled from traders and trappers were positive for coronavirus at the first node of the rat supply chain, but this proportion increased to 32% in large markets and even 56% in wildlife restaurants at the end of the supply chain. So we observed a clear amplification of the coronavirus infection along the wildlife supply chain, from 21% at the beginning of the chain to 56% at the end of the supply chain. So the high proportion of coronavirus positive samples at these human-wildlife interfaces highlight the high potential for human exposure to wildlife coronaviruses. And this viral amplification that we observed along the wildlife trade supply chain for human consumption highlights the potential for coronavirus shedding and amplification along other wildlife supply chains. So, for example, what we observed here for rodents could also happen for civets or pangolins, for example, which are hosts of SARS coronavirus-related viruses. And these species are heavily trafficked and raised in wildlife farms in Vietnam. So livestock and people living in close contact with rodents shedding these coronaviruses provide opportunities for intra and interspecies transmission and potential recombination of coronaviruses. So increasing viral surveillance at these human-wildlife interfaces is very important in the future in order to rapidly detect viruses with potentially a high zoonotic potential. So now I would like to thank my colleagues from EcoHealth Alliance and from China, with whom I conducted this study on bat coronaviruses in China, for their collaboration and support. And I also would like to thank my colleagues from WCS in Vietnam and other institutions in Vietnam who were involved in the study on rodent coronaviruses in Vietnam. Fourth, I also thank you for your attention, and I would be happy to answer any questions that you may have about these findings and about my presentation. Thank you.
Okay.
Thank you very much, Dr. Alice, for your very impressive data and impressive result, and both from bats and rodents. We have not so much time because we are a little in the program, but I have one question because you're online. For the bats, you present a lot of analysis looking at host switching events, and you did not discuss so much about recombination. So do we suspect that you have also recombination, especially in the alpha for the bats?
Yeah, can you hear me?
Yes, go, Alice.
So yes, we don't mention recombination in that study because the study is based on a short fragment of the genome, the RDRP gene. So we could not look at the whole genome for that study, but for sure, recombination occurs also among these viruses. But we could not look at that.
Yeah, when you look at the patterns of your two trees and that you discussed very well on the alpha and the beta, do you think that you have much more recombination into the alpha, potentially, or not?
Potentially, yes, but we cannot say that based on the data that we have, unfortunately. We would have to look at whole genomes for that, but that's something that could happen, yes.
Yeah, and there's a question from Joanne. If spillover are so difficult for beta coronavirus, how to explain the spillover from bats to humans? Especially
I know. It's and we looked at the macroevolution in bats, and when you look at that, it's clear that there are more cross-species transmission events for alpha than beta coronaviruses. But yes, when I know that the spillovers into humans were from beta coronaviruses. So the examples that we have are a bit against the findings of the study.
And there is another.
It could just be that these SARS coronavirus are really well adapted to humans and to other mammals.
Okay, yeah, okay. And the last question, because I think I'm very happy that Mariade could join. There is a question of, not a question, but a comment from Julian about the rodent wild farming, and that we cannot really understand that the murine coronavirus can go. But at the very beginning, you show that, especially again to the spillover of humans, is not going directly to humans. You have an intermediate host. So do you think that, okay, you have the increase of the coronavirus prevalence along the value, the wildlife value chain, but do you think that it's the same for the risk in terms of risk? So in terms of the real transmission to humans?
I don't know, but the exposure must be higher at the end of the chain than at the beginning because you have more positive rats. And at these interfaces, you also have a lot of livestock, for example, who could play the role of the intermediate host. And so the livestock at these interfaces will also have higher exposure to these rodent coronaviruses.
Okay, yes. Thank you very much, Alice. Thank you. And you have to go to the next speaker.
Okay, for the next speaker is Associate Professor Dr. Meredith Le Guillard, University of Creil, France. Her topic is discover an One Health approach to disentangle coronavirus evaluation and emergence.
Mariade, you online. We see you. You can share your Your PowerPoint. Maybe there is...
Yeah, I'm sharing.
Okay, thank you.
Thank you.
Alert references system. Cisco Weeks Meetings.app Naples.
Okay, so just a minute, that's okay now. I have to just reload the application. It will take just 30 seconds if you
Okay,
may
or
allow me to do
what
so,
we can
because
do, it will take
there
your
will
time
not
for
be
doing this.
recording otherwise.
Take your time to do this, and we can go to the next speaker because it's also already recorded, and back to you after. Okay?
Back
Thank
in
you.
30 seconds.
Okay, for the next speaker is Dr. Nicolas Linnet from IRD Museum National Historia Natural College de France. The topic is Farmer and Veterinary Knowledge towards Hybridization for Sustainable Farming Systems.
Good evening, everyone. I'm very happy to have the opportunity to present for this third seminar at KU. And so in the lens of the TICA project, and notably its traditional and veterinary knowledge aspect, I'm very happy to present you today some preliminary data collected in NAN. And this presentation will actually offer an overview of a multidisciplinary project recently launched in NAN, focusing on extensive buffalo farming. So, but before that, I will first focus on the context and the overall objective of this project. So, as you know, on a global scale, research have now highlighted clearly the importance of expanding livestock farming, both as a threat to biodiversity and as increasingly putting human and animal health at risk. However, as Serge has highlighted in a paper, little distinction is made between the different production systems from ranging from smallholder to industrial farm on the one side, and from the diversity and species distribution on the other. This last aspect is above all crucial, as it actually depends both on the various socioeconomic context and on the cultural value associated with animals in different cultures. So while the finding is really clear about the impact of livestock globally, the article better nuance and concludes by a call for further contributive study in order to better understand the complex links between farm production system, biodiversity, animals, and human health. And such a finding can directly be connected or linked with a recent and true provocative report published by PAS3, a European research program on pastoralism globally, entitled "Are Livestock Always Bad for the Planet?" Well, I believe the title speaks for itself. And focusing on climate change, which is directly linked with a recent and directly links to health and biodiversity issues, the author of the report offers an alternative to the dominant picture of livestock, a picture, they say, is often distorted by faulty assumptions, mainly based on intensive industrial farming in rich countries. So the author actually points the fact that extensive livestock farming is often excluded of the debate, and that the smallholder or family farm are often overlooked and not properly taken into consideration. So far, indeed, little attention has been paid to extensive pastoralist-like livestock system and their potential impact on maintaining a healthy socio-ecological system. And actually, while extensive ruminant-dominated system is often attributed with high greenhouse gas emission, taking a closer and another look at the picture, one can advocate of a low input grass-feed, and then grass-feed system can be presented as a paradigmatic sustainable system because of their biodiversity, land use, rural development, as well as animal welfare benefits. So the buffalo farm recently launched, which is then in its preliminary stage, goes in that direction and aims at exploring the various ethnological, botanical, religious, ecological, and medical dimension of extensive buffalo farming under One Health approach in order to seek for its potential multiple benefits. But first, we'll ask why the buffalo? Why? Well, actually, among the most common farm species found in South and Southeast Asia, chicken, pigs, or cow, buffalo occupy a special place in the bestiary, both for practical but also for religious purpose. In past, the animals were indispensable for paddy field cultivation, and at the same time, they were of religious and symbolic importance. For example, the annual buffalo sacrifice held to protect the whole community was of major importance to assure the health and well-being of everyone, human and animal. Obviously, most of this practice does not exist much nowadays. If you look at Thailand now, along with the Green Revolution, rapid changes, including modernization and mechanization, have led to the hand of mobilization of buffalo labor force in rice cultivation. Nevertheless, the animal have not totally disappeared from the rural landscapes. And for some local population, buffalo farming is even still very much present as the animal constitutes an important economic capital. And so where it still exists, the extensive buffalo farming system follow a seasonal activity directly linked to the agricultural calendar. It means that the animals spend about half of the year in the village area along the paddy field after the harvest, and then they are sent back to forest before rice transportation when the monsoon comes. So for the beef farm project, I will now briefly develop the specific approach. Because this project is primarily grounded in anthropology, as I'm a social anthropologist, and humanities, and will take ethnography as a sole method. And so one of the project objective is first to highlight, to unpack, what the French anthropologist Jean-Pierre Digard has called the domesticatory system, which result from the combination of biological, technical, and human factors that characterize the use of an animal in a given region. And so the particularity of the project is it's a strong participatory dimension. For the beef farm project, the local herder knowledge will be the starting point of investigation in each of its dimensions. On field, this herder knowledge will be documented in their relation to animals and to the environment. And more crucially, this knowledge will not be blocked or considered as a finished result, but more as a result of a dynamic process that must be apprehended in the course of action. And adopting such approach implies to take into account the fact that the object in which the knowledge is implemented is not an artifact or an inanimated object, but rather is a living being, endowed with the capacity to act. So for this, the project adopts also a disanthropocentric perspective. And what is interesting in adopting such a disanthropocentric or multi-species perspective is the fact that while conducting the fieldwork and data collection, invite not to choose between the human or the animal side, but to carefully look at the network of relation they build and share in their common environment. For example, and just an example, relying on this local knowledge, it is possible to explore the animals' exploitation of resources, following what anthropologist Florence Brunois has called ethnoethology, which is actually a local ethnoethology, of animals. And this means accessing to the knowledge and understanding of animals' environment through the mediation and interpretation of the herders themselves. So some of the idea and ultimate goal to develop will be to put this local knowledge into dialogue with the various scientific perspective, and as well as to highlight or to seek for virtues or good practice for livestock farming, guaranteeing the good health of human, animals, and the environment. So before I will move on and show how this approach will be, which has some anthropological, botanical, and ecological component will be developed, I will just introduce you to the field site, the village of Banwaipan in Nan. So Banwaipan is located along the Nan River, presently at the border of Tung Chang and Cheng Klai district of Nan province, but it administratively is attached to Cheng Klang district. Interestingly, it is a Lua village. It is not a old one, and those Lua village originated from Buklua district, and they escaped from the conflict in the 70s. And most of them at that time bring their buffalo along with them. Lua are basically hilly tribes of Thailand. And so today in Banwaipan, you have 180 inhabitants, and you have an approximate number of 250 or 300 buffalo. To give you another information about the specificity of the village, it composed of 52 households, and among them 47 possess buffalo. And probably for this reason, and in the will to develop the local population and development, the village has been selected to take part of a national buffalo breeding initiative. And last year, on this occasion, they received 30 extra animals. So as I mentioned earlier, the extensive buffalo farming is a seasonal activities which involve the annual circulation of animals from the village to the forest. And so here you have an approximate view of the field site, where you can see the village in blue, the culture, the paddy field, the tungna in yellow, and adjacent to this, the forest or the grazing area for the buffalo, the pankwai. And so, considering the seasonal movement and circulation of animal, the project imply to have a multi-scale approach, and as well as a multidisciplinary one, which means conducting research in the pankwai when the elephants are there, and in the village. Actually, it's simply to follow the seasonal migration of the animal. So, but first at the village level. So understanding the activities related to buffalo primarily means to conduct research on the daily relationship between herder and buffalo, and for that mobilize the method used in ethnozoology and ethnobotany, which aims at documenting the system of knowledge and representation of animals and plants. In concrete terms, this means taking an interest in any animal care and practice, and when necessary, carry out an in situ herbarium with identification and taxonomic survey of fauna and flora in the vernacular language. Obviously, the associate use and technique will be observed, as well as the interpretation of the animal feeding behavior according to the herder. One important aspect I could notice during my exploration was the presence of medical pluralism to take care of buffalo. Actually, one aspect of the project is to avoid to making a separation between the ethno-veterinary medicine and the contemporary veterinary biomedicine, but to consider them as a whole and as a system. As you can see, indeed, herders use both medicine in their daily activity. So I have started slowly conducting some interview with the herder. Sorry. Yes, interview with the herder first to document the buffalo diet and plants that the animals consume when being at the village level. I also was able to document some local medicine or local preparation, and for this one, the woman was preparing a mixture of 3 different plants to help a female buffalo who was just delivered, and the preparation will help her to recover from her pregnancy. And locally, as you know, also the medical practice are also associated with traditional beliefs and cosmology. That is to say that in addition to document the local medicine of plants, the project will also focus on what can be called the medicine of rituals, which is performed, for example, when the buffalo come back to the village in October, November, they will thank them, and also they will have to offer to the deity of the village. Another aspect, and very interesting at the village level, will be to assess the ecological role of the key cry or the buffalo poo, because during this dry season, the buffalo are free to roam in the paddy field or in a small adjacent forest area, but at night they are tied to specific plot or rice field where they will spend the night, and in the morning, as you can see, the herder comes and collect the poo, and they will distribute and disseminate it for paddy field regeneration. So I will move now to the second place, the pancreas, at the pancreas level, or the grazing area. So as I already mentioned, and the seasonally buffalo drive to the adjacent forest, which is located at the limit of the village and the culture area, where they will spend about 5 to 6 months from May, June to October, November. Actually, all the buffalo owner will decide together the appropriate date according to the progress of work at the tugna, at the paddy field. And prior to let their buffalo roam freely into the pancreas, a collective ritual ceremony is compulsory. The liang feed, which is really common in Thailand, feeding the spirit, is performed to ask the spirit of the land to take care of the animal during their seasonal stay. And it consists of offering a pair of chicken with also incense stick, candle, betel nut to seek protection. This period, indeed, is when the animal are sent to the pancreas is a very risky one, as it also occurred during the monsoon. One farmer confessed me that last year he lost 2 of his buffalo because they slipped on the ground. Another one told me that one of his buffalo died last year because his horns were blocked into bamboo. There, the buffalo are also exposed to wildlife encounter. And while the older farmer who settled first in Ban Wai Pan told me that when they in the 70s, a tiger was still present in the adjacent forest. Nowadays, they face the presence and attack of wolves or reptiles, which are still present. Nonetheless, in the pancreas, buffalo benefit from a larger space to roam, and at the same time, also this season, all buffalo share a common place, and so this period is also full of hope for the herder, because mating generally occurs during this period. And during this period, each owner will come and visit the herd and the animal and control for his own animal and for another animals, and to check if the buffalo are in good health and if nothing has happened to them, if they don't face any danger. So side of the Pancroy, at the Pancroy level, the project will develop a specific methodology of which first will aim at completing completing the buffalo database and also seek to explore self-medication practice. So the objective will first be to document the diet of the buffalo, assuming that in Pancroy, and especially during the monsoon, the animal have a larger choice of food. And this part will rely on a previous research I conducted on elephant in Laos, where the idea was to follow the animal along with their owner to observe the plant they eat, and then to seek for potential self-medication practice. In addition, some of the buffalo are probably the chief for each herd, because for each farm and each herd has a chief among them. They will be equipped with GPS collars so that it will be easier to track them and to see which part of the Pancroy they prefer to stay or not. Moreover, in order to assess the sanitarian risk, the interaction of buffalo and wild animal will also be observed. And for that, it is with the idea is to put some camera trap positioned in a strategical points of contact and interaction. And I'm particularly thinking about this the sacred place, the 2P, and the house of the spirit. And the hypothesis indeed is that such a sacred place are known by animals. Some offering are left also on site, and there are two crucial points or attractive points of encounter of animal, wild or domestic, with potential zoonotic risk. And interestingly, there exists a very rich anthropological literature on this sacred place across South and Southeast Asia. And obviously research only anthropologically focus on the symbolic and religious aspect of it, but little information or attention has been paid to what happened in practice, particularly in terms of interspecies exchange and encounter. In particular, the question will be what happens to the offering, who consumes them, because there'll be some rice and some chicken and some flour, some fruit, and etc. And who consumes them. So the idea will be to equip those seats with camera trap and to observe which animal will be visiting them. So the research conduct in the Pancroy promised to be ethnographically rich, but also methodologically challenging one for the use of GPS and the camera trap, and it will be, of course, necessary to complete and supplement the information collected on the buffalo diet at the village level. And so this will be the next step of the project, which will start during the next season, as you understood that all the buffalo, are currently in the village now, and they will move there by May or June. I have to get the contact with the herder and the Loa people there. So before I conclude, I would like to open the prospect and envisage the collaboration, because as you see, and I must say I'm very happy for having presented you what we can call the menu of the project. But everything has to be more has to be collected done, and as you can see, with the strong multidisciplinary dimension, I alone do not pretend to cover all of the aspects, and so after my initial exploration and preliminary data collection, in Banwaipan, back to Bangkok. Recently, I prospect for some collaboration. And there I was really lucky to meet some fellow, first at the International Buffalo Information Center, the HIBIC, which is hosted at KU at the library. And this is probably a unique center of exchange, distribution of information on buffalo between worldwide users. The center also possesses a huge literature on many aspects of buffalo farm in Thailand and elsewhere. And they also maintain an update database of buffalo in Thailand that they already share with me. So I'll be happy to be associate or to associate HIBIC for this project. Side of the plants and the local medicine, I was also able to visit the Bangkok Herbarium from the Ministry of Agriculture, and the herbarium is located also on the KU campus on the side of administrative unit, and some formal collaboration are currently being established to make a deposit of the plants and voucher, and already few specimens have been deposited and are waiting for official agreement to proceed. That will come very, very soon. Last, and I would like to thank him, Kim Fonupatich, which is a consultant botanist I was in contact with, as contact a lecturer at the from the environmental department, which will be interested in the study, especially to cover the environmental assessment impact, the EIE of the project, to understand the role of the ecological role of this extensive farming system. So thank you very much for your attention. I'll be very happy to receive any comments, questions, or if you find any missing aspect of the project, I will be also very happy to hear from you. Thank you very much.
Thank you very much, Dr. Nicolas. I'm very impressed also because you arrive and in 1 month, 2 months, I think you did all of this. It's very impressive, very impressive. I don't know if you are online or not. I'm not sure. No, you don't online. But what we will do is we can collect some of questions, because sure there will be questions for you, and we will go back to you. And we can go maybe now back to Mariadec.
Okay.
Please.
For next speaker is Associate Professor Dr. Moledex Le Gualert, University of Caen, France. He is going to talk about the topic Discover a One Health approach to disentangent coronavirus evaluation and emergence.
Thank you very much for this, giving me this second chance of sharing and presenting this content to you. So I will try now to go for the presentation, and it should be okay. Yes, could you see the screen?
Perfect. Perfect.
Okay, very good. Okay, very good. Thank you. So I will be introducing you this project, Discover a One Health approach to disentangle coronavirus evolution and emergence. Before this, and I would like to thank the organizers for the opportunity to present, and I will just make a quick reminder on the coronavirus. Just as everyone know now, probably these coronaviruses look like this particle with a round-shaped corona shape around the particle, and it could be also seen as these particles in groups with exhibiting these crown-like proteins, the spikes proteins you probably heard about during the SARS-CoV-2 pandemic. We will be coronavirus are divided in different genera, 4 genera: the alpha, beta, gamma, and delta coronaviruses, and they're have a wide host spectrum. Many different species can be infected by different species of coronaviruses, and they mainly give some respiratory and enteric diseases. And as you can see, we have also, as human, several coronaviruses since quite a while, let's say centuries. I will come back to this later. And we have these coronaviruses infecting human, both in beta coronavirus and in alpha coronaviruses. If we make a quick zoom on these beta coronaviruses, we have several coronavirus, seasonal coronavirus infect human. I just mentioned these two alpha coronaviruses in here in 229E and NL63 in the alpha coronavirus group, and other two HKU1 and OC43 that are seasonal coronavirus. And this is important. We will see why in the later step. Of course, you heard about more recently that we got other coronavirus, such as the SARS-CoV that emerged in 2002-3, and also we got infected as human some part in the world, like the Arabic Peninsula. We got infected by this MERS-CoV coronavirus linked to camels, and more recently, of course, the SARS-CoV-2 that emerged also from beta coronavirus sarbecovirus group. So that's make many different coronaviruses, and just a quick word on the taxonomy of these viruses. So they're all from the nidoviral order in the Coronaviridae family, and the Orthocoronavirinae subfamily. We will be mostly focusing today on beta coronavirus, and in beta coronavirus, we'll be focusing mostly on sarbecovirus. I will just explain you what that means. Another broad range view of the diversity, of the genetic diversity, you can find in the database, in the genetic database. You have more than 200,000 sequences of coronaviruses today. The red one here is the beta coronavirus that has been most sequenced to date because of the SARS-CoV-2, because of the SARS-CoV and the MERS-CoV outbreaks, and also because some, like the OC43, has been much more investigated as the HKU1, these two seasonal coronaviruses. But you have also a huge diversity, less known, but a huge diversity in the gamma coronavirus and delta coronavirus, mostly infecting wildlife animals and domestic animals. So many of these coronavirus actually have impact in veterinary settings, infecting, for example, feline canine groups, or for the infectious bronchitis, the avian species, and also porcines and many and cattle as well for the another beta coronavirus. If you look at the genetic structure of the genome, they are all quite similar, with many differences, but mostly similar in their organization, with a big part at the beginning of the genome, 20 kbs, 20,000 nucleotides for the protein complex, for the polymerase, for replicating the genome. And then you have the structural, what we call the structural proteins here in green and blue, the S, the spike protein, the membrane protein, the nucleoprotein, which is actually packing the genome inside the particle, and also different accessory proteins for some that you can find only in some groups, like the enigmatic HE protein, hemagglutinin esterase, that you find only in some beta coronaviruses. So all this protein packed together to form this viral particle. Just to summarize these coronaviruses, we have the historical coronavirus, an alpha and beta, and 229E, and the OC43, known approximately in the 1960-70. The new coronaviruses were discovered, also seasonal, also give also flu-like syndrome, and seasonal in 2004-2005, after the emergence of the most famous ones, like the SARS-CoV in 2002-3, the MERS-CoV in 2012, and the SARS-CoV-2 in 2019. So these last ones were the truly emerging coronaviruses we much know and we much heard about the last years. But if we look at the impact of this coronavirus, mostly the seasonal one, I mean the four first I mentioned, the seasonal ones that commonly infect people in winter, they're responsible for approximately 10% of the flu-like syndrome in winter, and in mostly infecting people in childhood before, let's say, 30 months. Everyone has encountered at least one or two coronaviruses of these seasonal coronaviruses, and you see here as time goes by, up to 12, 24 years old, you met many times, and quite all the population has met the HCOV OC43, HKU1, and L63, 229E, these four seasonal coronaviruses. So they are quite frequent. They circulate widely, they circulate broadly, they are present everywhere, and they infect everyone approximately before the age of 5 years old. And then recurrently over the life, they come back to infect the same people. So what about the timing of emergence of these viruses? And this might be very interesting to take in account when you look for the emergence of the actual emerging coronaviruses, or if you want to look for the next ones. The NL63 emerged probably 700 years ago, the 229E like 200 years ago probably, and the OC43 that came from the bovine coronavirus, a domestic animal, emerged in about 120 years ago, and the HKU1 emerged most probably in the 1950s. So we've got many coronaviruses that emerged during the recent history, and this is only the one we know, but we probably have other emergences that has not led to a real establishment in the world population. If we just try to summarize these emergencies, and we all heard about the implication of different animals, from wildlife or bats or even other animals. So what we can say today, even some hypotheses need to be much more investigated. But we have, for some of them, relatives in bats. That's now clear, with sometimes the involvement of probably an intermediary host. For example, for the SARS-CoV, you remember maybe the story of the civet cat. I will tell you again in a few slides. There has been the story of the SARS-CoV as well that emerged in porcine, and that was a big problem for pig industry in China and US. You have this, but that does not emerge in human. You had the story of the MERS-CoV that use, let's say, the circulation in the camels in the Arabic Peninsula for 30, 40 years before infecting human. You have also the 229E, which is probably, maybe, the origin is not clear, the intermediate is not clear. And SARS-CoV-2, for which, of course, the intermediary host is also not clear, but for which the story of emergence, I will explain this, is really, really reminding the story of emergence of the SARS-CoV. And you have also the HCOV and the 63, for which we have several relatives in bats, but for which we lack an intermediary host, because this is quite, you remember maybe this is the most ancient emergent coronavirus that emerged several centuries ago, so it's difficult to trace back. And the OC43 that clearly comes from cattle and about a century ago, a little bit more than a century, and the HCOV HKU1 that for which we found some relatives in rodents, but for which the intermediate host or the relation of the story of emergence is still unclear. So if we switch to now what we've been doing, we will be doing in Discover projects, it's linked to what we've been doing in Europe, and that will be the same approach. That is the same approach that is used. So just estimates diversity. This is a project called Epicurium we got a few years ago, and for which we used the diversity we detected in Europe. But I will just focus today, as I told you, on the sarbecovirus in a specific species of bat, Rhinolophidae bats, the Rhinolophus pharomequinum bat. So after the screening and of many animals, and the animals of interest, as the host of interest has been found, and the sarbecovirus has been found in a colony of bats in Brittany, we've been following this colony for several years, and this is this result and approach I will describe now. So, and this also has been a little bit investigated in Thailand by our group and by others, but this was more than 10 years ago. So I don't come back much about this, but this has been also started years and years ago. So a quick reminder of the SARS-CoV hypothesis of emergence, because as I told you, this SARS-CoV-2 history might be very similar to what happened with this SARS-CoV. We had a diversity of coronavirus circulating in wildlife and also in bats, in Rhinolophids bats, with many different species. In Asia, I'm sure that PPAT showed you how diversified this Rhinolophus can be, and they harbor many different coronaviruses. By chance, some of these coronaviruses, and by the process of evolution, pressure, selective pressure, and diversity, these coronavirus, some of them can, by chance, jump and infect another host, for example, civet cat, Paguma larvata. This is called Paguma larvata. And this species is also by chance for the virus, a species that has been breeded by human in South China. And the breeding facilities have really exploded in the few years preceding the emergence of SARS-CoV. So that might have been an explanation of the starter of the epidemic and the trigger of the epidemic that promote the virus to come from the wildlife, and by the play of changing this density of animals by farming them and by really promoting the spillover and transmission between these individuals and animals, has made a kind of reactor in which the virus was really circulating. And this has been observed in Europe as well for the SARS-CoV-2 in the mink farm facilities. Even in France, we observed this. We've been investigating some mink farm in the aftermath of the SARS-CoV-2 emergence, and we've seen that the virus can circulate in this typical environment of farmed animals without any sign for the people that take care of them. So it was not possible to see the virus, to see the animal sick, but the virus was really circulating in the animals and evolving inside the farming facilities. And we got the evidence that there has been infection between human and animals at this interface between farmed animals and humans. So this is a typical example of a zoonotic infection, and this has probably happened for SARS-CoV, and this happened again for the SARS-CoV-2. The point is, we don't know how, and we don't know the really origin, and we don't know what happened exactly at these steps. And that is a very burning question, actually. And then for the rest of the story, it's more easy because you got already a virus that is adapted to humans. So if you got this virus adapted to humans, and if this virus reach a highly density, highly populated area, then you get all the chances for this virus to expand, and you experience it now since 2 years nearly. So on the principle, that was what was happening for the SARS-CoV-2. for the SARS-CoV probably, and probably also for the SARS-CoV-2. And this rely on the most ancient idea that in the ecosystem, humans live in ecosystem. And we have this frequency of pathogens of microorganisms, not necessarily pathogens at this step, that fluctuate in nature, that circulate naturally, but never really get a point in which they cause a big epidemic within animals, because diversified environment really promote the dilution of all these pathogenic elements and doesn't lead for pressure, selective pressure reason, doesn't lead to big pandemic or big epidemic. But if one of these animals, and if the condition change, and this animal by chance encounter either a domestic species or another animals that is more densely populated, for example, this virus can develop and increase and invade this new population of hosts, especially if they are domestic animals and if their genetic is quite homogeneous and if they can be actually in high densities in some places where humans also are not far, and they may also spill over to human. And this is a typical simplistic, of course, but quite interesting for understanding a model of emergence. And some have hypothesized that if we are ready to make enough forecasting or early detection, which is by itself good, it's always good to know what is actually there. And we could maybe put in place control operation. And this is more tricky, to my opinion, because it's very difficult to control it, and it's always the idea to control the biodiversity, and it's always the idea, ancient idea too, that human can control everything. So I'm not sure we could really when we got this early detection and forecasting and understanding of all the steps, make really the point to make control of the biodiversity in the microorganisms. So we may have also to rethink globally and to see how the ecosystem by themselves, their settings, the fact that they're diversified and the biodiversity they have maybe may provide a buffer role and avoiding for these microorganisms to take too much space and in the animal world, and finally maybe also in the human world. So this is the idea, to see if there is some what happening in this project is to see what happening between this fluctuation of microorganisms in wildlife and what happening between this fluctuation and what actually happens in domestics or wildlife or humans. This just I want to expand on this because I'm sure people has explained you everything about diversity. If we speak about the model we've been developing is on the Rhinolophus equinum. We've been looking, of course, about this coronavirus, and this is a species that is widely distributed along Europe and also to Asia. So in these colonies, we've been sampling, and this is the methodology we would like to apply in Discover. We've been sampling frequently these colonies, this maternity colony. You see the young babies here and the mother here, and the mother with the young babies. This one is trying to fly. It's really nearly the time for them to leave the winter roost, and they live all together. They, of course, have close contact. So a coronavirus, a sarbecovirus there can really be effectively circulating in the colony. And this is exactly, this is another species from Thailand. But this is exactly what we see in the colony. When we sequence the whole genome of this sarbecovirus within one colony over time, we see that this virus is actually changing quite fast from the year 2014 through 2015, and then 2016, 2017, and we got the data up to 2021 now. We see that the mean rate of evolution is 10⁻³ which means this amount of substitution per site per year, which is quite high. It's the highest observed for coronaviruses. Another point in this picture is when you look at the year 2016, for example, you see that there is a gradual evolution of the virus from the beginning of the year to the season to the end of the season. So, and if you calculate the time in which you change a virus get replaced by another one, let's say like this, or what we call the variants with the SARS-CoV-2, for example. If you calculate the time necessary for a new virus to emerge in the colony, it took approximately 2 to 3 months to get a new virus in the colony, to get a new virus that replace the old one in the colony. So this is based on the whole genome sequencing, but you could also, and this is a comparison for what we've been seeing with SARS-CoV-2 in the human population in the last year based on our data. And this is the mutation rate. The mean rate is much lower. It's 10 minus 4. Before it was in the bat colony, it was 10 minus 3. So it evolved quicker. If you compare with other seasonal coronavirus, you got this 10 minus 4 rate of evolution. For example, for this NL63, we have been following for more than 10 years now. So this evolution rate are different in wildlife and in humans. So it might be linked to the way the virus circulates and to the pressure the virus is facing to within these different host settings. Moreover, if we have a quick look at the evolution rate is quite constant. So the changing of the virus it's changing at the same rate over time from 2014 to 2017. The changing is linear. So and if you look now out of this main genome and the whole genome analysis, if you look at the intra-host diversity by next-gen sequencing, if you look at the diversity inside a given individual and you try to map all this diversity along the genome in different individuals, you can see that you have a very difficult to interpret a pattern of pressure, selective pressure. So the selective pressure is actually acting differently in different individuals in different parts of the genome of the virus. So this is really something we have to investigate to be able to understand how the virus is reacting to the host ecology. More broadly, if you just consider the number of individuals infected in a colony at a given time, at a given moment, and if you repeat the experiment all over the period from 2014 to 2020, each year you have several samples of the same colony. So let's say hundreds to 500, 600 individuals sampled per year or per season in the same colony. You can see that for the time where we have enough sampling, we have a high amount of individuals infected, nearly 100% sometime at the beginning of the season, of the spring. Then it drops down in a few months to get really down to nearly 0.5% of individuals infected in, let's say, beginning of July, and which is actually the period where the virus, the individuals and the litter is growing up and stopping feeding on the female. And then just a few months after, you have again, after the litter is let's say feeding by themselves and not protected by probably by the maternal immunity through lactation, they got their own infection, and they promote the reinfection of the colony by this virus, or probably by a new one, by a new variant. And then this rise of the prevalence in the colony is also getting a second peak, let's say 3 months later, after this individual, this new litter has been infected. And before after this stage before starting again to rise. But at this winter period, we have less points, so we are less confident with our data. But we have this kind of bimodal evolution of the prevalence of the colony, like if they have 2 epidemics per season in the colony. It's going quite fast. This wave we are describing now with the SARS-CoV-2 in humans are actually also occurring in wildlife, even in very small settings like this this colony of Rhinolophus perimecrinum. And if you compare with the data on SARS-CoV, which are of course very diversified now, we look at the diversity of the epidemic based on our data sets, more than 10,000 samples, and we have actually a correlation between the wave, the peak of the wave, so the amount of circulation of the virus, the number of people infected, and the diversity, the genetic diversity within the genomes. And so for the first wave in March 2020, we have a peak also of genetic diversity, and for this another wave in September 2020, we have another peak of genetic diversity. The wave is the biggest. The diversity is in the circulation of the virus. So the intensity of circulation is directly linked to the diversity of the virus and promote its ability to change and to adapt to the next generation and to the new host eventually. So if you have big waves of these coronaviruses in a given place at a given time, and if you have at the same time the encounter of a different host, you have a maximum probability of emerging a new lineage.
Sorry, Meriadec, you have to conclude. Yeah. You already speak 30 minutes.
Okay. Sorry to be a little bit long. So I just go to the end. These other pictures are just saying that the diversity is not actually the same in any place in the genome, and the pressure, also the selective pressure, is different when you look at different proteins of the virus. If we come back to the SARS-CoV and the SARS-CoV-2 origin, we got some clues that this is a mosaic of viruses, and actually a mosaic that means that many viruses has been interacting together, exchanging genetic materials to build the actual genome of the ancestral genome of the SARS-CoV. Before the SARS-CoV-2 emergence, we already knew from China a huge diversity of sarbecoviruses, much less from the SARS-CoV-2 group, and a little bit, of course, less in Europe, for example, but we have also. And what would be interesting is to know better the diversity of SARS-CoV-2 coronavirus that circulates in nature, and to better estimate the time of the evolution and the time of emergence and separation of these different lineages. So for this, we come back to this SARS-CoV-2 model, and what we would like to really, that you know now perfectly, what we would like now is to really assess the origin and natural history of new lineages. Not specifically SARS-CoV-2, because it's probably too late, but for other related viruses, to see how these viruses actually interact with wildlife, and how the diversity from wildlife has an impact in the coronavirus evolution to infect other animals in the vicinity, wildlife or domestic animals. So this would be very crucial to study. And with this approach of concentric circles from wildlife, from the hotspots, from the diversity of these viruses, and how we see this diversity changing when these viruses reach a different layer in the ecology. And of course, all this following a diversity ecology gradient from wildlife to more domestic or anthropogenic environment. So this is the main objectives is to estimate prevalence and diversity of sarbecovirus, to see the temporal evolution of this virus in wildlife, and see the interspecies spillover by this method of sampling by concentric circles around these colonies, and to see the evolution of the SARS-CoV-2. And then we could, with all this data, estimate on a temporal approach and a high frequency sampling approach, we could estimate the zoonotic potential and of this different virus in linked with the environment, the genetics, and the socio-ecological factors that are actually recorded together. So thank you to, I would like to finish by thanks to all collaborators, of course, Serge Morin, Pipat, Nose Song, and many others that I don't remember the names, but and also to the Connected Project. And I thank you all for your attention.
Thank you very much, Dr. Meriadec. I'm sorry, because we have to really to finalize at 7 o'clock, and people are leaving after. I'm closing the one. Very interesting talk, very fascinating. I think you will need all the afternoon, and for only for you, only so very interesting. So we wait for you next year to come, and we will give you a whole afternoon to teach. I think it's also interesting. And we will go to the last presentation. Thank you. I'm sorry again to continue. Thank you.
Okay, from the last speaker is Dr. Claire Jennings from INSERM LEPD, University of Aix-Marseille, France. The topic of our last speaker is One Health, Policy and the Law.
Hello, everyone. I'm very happy to be invited to this international seminar, and I would like to thank the organizers at VetTech University for this seminar. And I'm Claire Lajoni. I'm a researcher at INSERM, the French National Institute on Health and Medical Research. And I'm a researcher in environmental law. And I would like to present an overview of the law regulating wildlife in Thailand and beyond, that is to say, internationally and in Southeast Asia. So first of all, what I want to present is the wildlife share comparing to livestock and humans. So there is today on the planet 1% of wild animals compared to 67% of livestock and 32% of humans. And there is trade of wildlife, whether it is legal or illegal, and it's at a very big scale, so it's very, very important. And it can be used for food, for medicine, traditional medicine, for pets, or for decoration or trophies. So it's an issue, and scientists warned the humanity, the planet, on the danger of illegal wildlife trade or even on unsustainable trade. And they insisted on the impact on different things, but on disease transmission to human, which is a major concern, and it appeared with the pandemic. The program of the United Nations for the Environment insisted on different factors, increasing zoonotic emergence in relation to the environment, and they notably insisted on the part of the livestock production, intensive livestock production, and the part of illegal or poorly regulated wildlife trade. And here is an example of mink farming all over the world. And mink farming is doing the link between intensive livestock production and wildlife. So here there is, of course, an international framework of law on wildlife trade. This is the CITES, that is to say the Convention on International Trade in Endangered Species of Wild Fauna and Flora. Here we will insist on animal. And its role is to ensure that international trade in specimens of wild animals and plants does not threaten the survival of the species. But the main issues that has been highlighted, and even recently, is that the illegal trade of wildlife and timber, because they are often associated, is embedded in the legitimate trade, and it's difficult to detect the illegal trade. And this is the case here in Indonesia, where there is a high level of legal wildlife trade, but it is probably hiding illegal trade as well. So there is this international law, but there are many difficulties of implementation. First, because of the increase of animal trade on the internet. Here, this is an example of trade in Thailand, but of course not only. And Southeast Asia is at the heart of wildlife trade, and with demand from all over the world and all the continents. So when we speak about wildlife law in Thailand, what is important to have in mind is that there are in fact many different laws. So there is something which is coming from the constitution, so the constitutional law. There are different laws and regulation implementing the law, but they are at different level. It can be environmental law when it concerns Elephant Tusk Ivory Act, for instance, or conservation and protection of wild animal, or national parks, or national reserved forests. It can be customs law, and of course the part of customs is important because customs are regulating the entrance or the exportation of wildlife at the border. It can be criminal law expressed in different way, like in the Firearm and Ammunition Act. It can be in the Anti-Money Laundering Act or Criminal Code, so in different areas. And here is also in the law to fight corruption. So there are limits to Thai legislation that have been underlined by association. For instance, the Thai Society for the Prevention of Cruelty to Animals. And for example, the Animal Epidemics Act is interesting, but is limiting the definition of animal to only certain animals and not wildlife in general, as well as the Cruelty Prevention and Welfare of Animal Act, which is in fact concerning certain animal, but does not include wildlife. But this is not a problem only of Thai legislation, it's something that shows the limit of definition in the law. And wildlife law can be broadened in a One Health context, and it has been underlined by the UN report saying that wildlife crime is putting environment and health at risk. And so the International Organization for Animal Health decided a framework to consider wildlife health among the animal health, and to organize a connection and coordination of the surveillance system, for instance, and the organization combating drug and crime at the UN level also addressed the issue of preventing future pandemics by combating wildlife crime. And it insisted on the fact that the wild animals, it's the trade of wild animals that is allowing the circulation of pathogens between animals and humans. So here there is a concern that emerged for a long time, but that has been seen during the COVID pandemic. It's a call from John Scanlon, who is the former head of the CITES, the Convention on Endangered Species, insisting on the fact that wildlife must be protected from crime and trade for the sake of public and planetary health. So this is interesting because he's now insisting on the fact that we must go further in CITES. And there is a global initiative to address the gaps in international law and to have higher sentences given by the Court of Justice to prevent and to end wildlife crime. So here there is a coalition. It was just to show that there is a world coalition against trafficking crime, and many organizations around the world are working together. This is what we will see next. So at the regional level in Southeast Asia, the organizations working together are mainly the customs of the different countries and the Organization of Customs, Interpol, and the United Nations Office on Drugs and Crime, the ASEAN Working Group on CITES and the Wildlife Enforcement, and the intention, and it happened with the COVID crisis, is to enhance the cooperation within the actors of the region, the ASEAN, to address wildlife trafficking. And there is an ongoing operation called Mekong Dragon 3, and the point of these different organizations is to connect, to share, to target, to prevent, and to intercept the criminals doing wildlife trafficking. So now this is the time for ASEAN to ban wildlife trade for goods. This is a title that has been published last year, and there is a movement defending the fact that we should ban wildlife trade, not only illegal trade, but also legal trade. So we have two movements, the fight against illegal trade, but also some people willing to ban completely wildlife trade, which is a problem for the populations because it's a source of revenue, but this is something to be discussed. And Thailand is now aiming at fighting wildlife trade because of the risk of pandemics. So that's why there is a new concern about wildlife trade because of its importance and because the reason of zoonotic disease is also a reason to combat more wildlife trade. So they are all connected. And this new movement has an effect. Here, for example, in Vietnam, we can see that there is a big fine for trading rhino horn and a sentence. This is the longest sentence ever given in a Vietnamese court. this is 14 years in jail. This is the same with a tycoon that has been sentenced for poaching in Thailand, as some of you probably know. And here the point is for the Supreme Court is to set an example and to put a higher standard on the protection of wildlife. So these are examples, but they are significant at a period when there is an attempt to combat wildlife crime together at the regional level. One point is interesting in the legislation. The legislation, of course, all alone is not efficient. To be efficient, the legislation must go with a system of judiciary and prosecution and of cooperation, as we have seen, between the police, between the justice, to allow the international cooperation in criminal matters. There is also a need of enforcement. So we need all the different sectors working together: customs, the border control, the police, the police, and different enforcement agencies. Data, because data are necessary to monitor the wildlife, to see what's happening, and because the criminals are inventing new means of trading. We have seen the impact of the internet recently, and the driver and prevention, mainly the awareness raising and all the training, even of children at school, for example, are a way to raise awareness of the danger of wildlife crime and the need for protection of animals in general. So there are campaigns that you have probably seen. Some are with movie stars, some are with comics for children, but the point is to make people think about the importance of fighting wildlife trade, and when going to the market, consider the impact of what we can do when we buy something. And the impact of tourism has been underlined on wildlife trade, and it's huge. So this is something to consider in the advertisement or in the campaign for wildlife. And I would like to thank the assistant researchers working with me on the Future Healthy project, because they didn't work on wildlife trade first, but on different national strategies in Thailand. But I had questions and I asked them, and the discussions were very rich, and it helped me to think about how to present wildlife legislation today. Thank you very much for your attention.
Thank you very much. Very interesting talk, Claire, if you are there. I'm pretty sure we have time for questions because your talk was a little shorter. So be free. We're waiting for questions in the chat.
Around.
No question. So I have to ask questions. So my question, because you focus on the illegal wildlife, but today there was an interesting talk by Alice, and Dr. Alice, she saw that the important way is along the value chain, and especially for the rodents, which may be also collected, but maybe also for the farming, there is an increasing of prevalence of some potential pathogens that may spill over to humans and to livestock. And we know that some of these rodents, they are not of any conservation concern. They are not on the CITES list, they are not on the red list. So what do you think about finally the link between the One Health approach and the wildlife conservation and the law?
Hello, I hope you can hear me now because I'm not sure. I can put the video on, I don't know. Yeah, thank you for the question. So first, yes, I insisted on illegal wildlife crime, but there are now two movements and I want to insist on that. The first movement is to fight illegal crime, but the other one, and this is a strange one, I think, is to even ban the legal wildlife trade and the markets. And this is something which seems a bit, that's why I said there is a debate and we can debate that. And this is probably very difficult to ban all the wildlife trade, of course. And when people are saying that, you're really right, because I don't think they consider rodents as a source of legal wildlife trade, but they are more focusing on other products that are considered like for decoration, as we said, and that are legal. And something in the legal world, there are many questions between the legal and illegal considerations. And this is probably something the One Health approach can help to understand, because in fact there is a recent paper written by lawyers but also ecologists in conservation biology, I think. And the interest of the paper is to highlight the fact that the sentences, the fines, and the conception of the crime, so the difference between legal and illegal trade are very, very different according to the different countries. So here, this is the first difficulty of having a common approach of illegal crime, because the definition is completely different in the different even national legislation. So this is the first thing. And then probably that's why I presented it like that, but you're right, I should have insisted on the legal trade as well. Probably one thing is to through the research, because this is about innovative animal health, through the research and the results of the researchers to how can I say, to put the debate on the table with the policymakers. You cannot hear me? I don't know what to do. Is that okay? But I cannot hear you now. So.
You can restart. Yeah.
Okay. Sorry, from the beginning?
No, just when you were mute.
Because I don't know when it was. Sorry, because I didn't see the difference here. So.
So you can start the last your last paragraph. Yeah.
So I don't know. I was saying that probably the One Health approach. So I don't know if this is where you lost me, but the One Health approach I think is interesting to reconciliate the legislation on illegal and legal trade. Did you hear the part on the paper on conservation, the legal paper I was speaking about?
Yeah.
Okay. So I think from that, because the cooperation is difficult between the countries because of the difference in legislation, the fact to highlight the One Health approach and to integrate the results of researchers, of scientists into the legal debate is probably crucial, and this is exactly the right time to do that. And the value chain approach is the approach of the International Organization for Animal Health, and it's probably something to consider. And this is the point of a project proposal that we will submit next week, hopefully, and to determine what is happening in the ASEAN region and what kind of connections or adjustments between the different legislation at the national level can be made to have the same kind of sentences or level of punishment for wildlife crime, but also to treat the legal trade of wildlife. So both are connected, and this is the connection that is rarely made between illegal and legal, and to treat also the animals, as you said, and we have seen that before, that are not considered as part of the conservation protection because they are rodents or they are not useful. This is not my point of view. I'm saying that because people are saying that useful or whatever. So they are not considered, or they are under considered, and probably this is to put into the debate. This is the role of researchers when they are meeting policymakers, probably, and this is probably something to add. There is a call here from me because I had the chance to discuss in the arena of the Convention on Biodiversity. This is probably something to say out loud, sorry, to people working on the link between biodiversity and health at the level of the Convention on Biodiversity for the next conference of the parties.
Thank you very much. Thank you, Claire. And I think it's time to conclude our session. So because we are at the end and we are just in time to conclude. And first, I would like to thank very much Dr. Vetinam for these 2 days, and we are just finishing this day. And for this day, to thank Dr. Anamika and Song, that was very important to make the link. And sometimes it was difficult because everybody was busy, and Song was really helpful to make this link. And I will thank to all of our speakers. So really, even we had sometimes some problems, technical problems or whatever, but I would like to thank all of you for all of the speakers for the really great talks, and we are very happy that some are already recorded, but this I hope you can use it, and we will ask you your consent to use it for the next. And for us, it's very important for today because this was the second year of the innovative animal health supported by the Thailand International Cooperation Agency. And really, we are on the track for the expected outputs of the project. First, in terms of innovative research that aims at maximizing, as I say, biodiversity and health, co-benefits for human, animal, and environmental health. And really, a lot of the talks today are really on this way, and to promote actively the participation of community. This was the case. Community science. This was the case on local expert. Really also happy for the second expected output with Nicolas, Dr. Nicolas, for the innovative research in traditional veterinary knowledge for pharmacopoeia. And I think beef farm are incredible for this way, starting very we are really happy to have this ongoing. And the third is really the development of this operational One Health framework. And I would like to say that we are also on track with this. We are on track because, of course, we have all of these networks, like GRIS network, like the other network. But now we have also PRISOD, the PRISOD initiative, to start this preventing zoonotic disease emergence that is supported by the French INRAE, IRD, and CIRAD for this, and they are finishing all of these workshop, and we will have soon some key elements for the next year's for news project, and we have also some very new project that can start on this. We have also all of these international and regional perspective and I will say expert panels on the One Health, with also related to the international organization, the World Health Organization, the Food and Agriculture Organization, the World Organization for Animal Health, and the United Nations Environmental Program. All of this is really to better put together sectors and disciplines to build a One Health and to better coordinate and communicate with society. And I think our project on today, we are really contributing to this big international effort. And finally, the last expected output, it was to because we are international expert hosted by the Thailand, hosted by Casings University and by Faculty of Tropical Medicine. We have to participate to strengthening Thailand research and academic capacity, really to fulfill the Thailand 4.0 and to put the Thai university at a better ranking with innovative tools. And I think that because we are collaborate, we are contribute all together, we are building new project, we are publish high-level citation papers, we are really also on track for this important expected outcome. So I finished, and I thank really very much all of you. And I thank again the Faculty of the Faculty of Veterinary Technology and Dean Professor Dr. Satapom, and also the President of Casings University. And it's time to close. Thank you very much.
Thank you, Serge. Thank you. Thank you.
Thank you very much to all. Thank you to you. Thank you.
Okay, thank you, Professor Dr. Serge. Thank you, everyone, for being here today. Tomorrow will be the theme Animal Health and the Environment. See you tomorrow.
See you
Bye
tomorrow.
bye.
Thank you very much.
ค่ะ
Thank you ครับ Bye bye.
Bye.
Bye, everyone. Thank you.
Thank you.
Thank you very much. Goodbye.
Bye-bye.
This is my voice one day learning in baking.
Thank you. It was okay. Yes, a little problem, but tomorrow I'm very happy to walk away. You know, when I find okay. starting to be too fast. They did not really. It's good that I don't take the plane. It's okay. Okay, thank you very much again. Sorry. Waiting for January. I'm beginning.